Good morning, Dougal.
Good morning.
So I might just introduce our dive for this morning. We're broadcasting live from the ROV Falkor in the outer waters of the Great Barrier Marine Park. Just arrived to this area north of Cairns overnight, and this is our dive number 378, part of our 4-week voyage exploring the seamounts, reefs and canyons of the Coral Sea and Great Barrier Marine Park. My name is Scott Nichol. I'm the Chief Scientist on Leg 2. And what you're just looking at now is the initial descent of ROV SuBastian, a site which is located in the waters of a large submarine canyon which extends from the edge of the Great Barrier Reef. I'll say more about the later part of the dive later on as we go down, but I don't want to give out our guest this morning, Dr. Dougal Lindsay, who's joining us from Yokohama in Japan, from his home. We go down the initial dive depths, the water column. We're going to be exploring and looking at the jellyfish in particular. Dougal is a world expert in jellyfish species, and I might pass over to you, Dougal, to tell us about the one we're looking at right now that's flashing on screen.
Yes, so this looks like, I'm pretty sure I saw a couple of filaments coming out of those triangular projections at the back end of the animal, which would make this Euramphia vexilligera. Right now there's only one recognized species, although back at the turn of the century, late 1800s, early 1900s, there were multiple species that were since all synonymized into the one. What we're finding more and more as we do ROV surveys is that because we can see the animals in pristine condition, not damaged by the nets in which they would have been caught in around the turn of the century, we can see morphological differences that suggest that sometimes those original descriptions were more accurate than the subsequent ones in the mid-1900s. Thanks for that. That video was recorded in a very high-quality video codec format that will allow us to go back and look at it in the lab. We'll probably spend a while going frame by frame to find in focus all the little pieces we need to see to give that a proper species ID. But I'm pretty sure that is very nice first observation. As we're driving here in our deep descent to the seafloor, this is a marine snow particle, not living. Looks like there's all sorts of things attached to it. Probably feeding on it. These marine snow particles, as they sink towards the seafloor, provide valuable food for the plankton and the organisms. Looks like a mucoidal particle. During some of our dives, we've been seeing what looked like it could be mucus excreted by corals, flowing out laterally towards the area where we're doing our dives, providing food for the midwater animals. And sometimes they're the houses of, here we go. Could be a piracy. No, it's a Burroi Foscali. Oh, yes.
Look at that move.
This guy is a fast swimmer. It's a Ziploc bag with covered in little paddles that help it swim. It can open its mouth humongously and eat things that are 4 times as large as this. The internal surface of the mouth in this species is covered with microcilia, so very long hair-like structures, which should be in lines running from the mouth down towards the gut. And these macrocilia are kind of like the velcros you have on your shoes. It's called magic tape, I think. So they're little gelatinous hooks that hook into each other and allow the mouth to close like a Ziploc bag when it's digesting something. And then it unzips from one side, opens the mouth up to get rid of the pieces it's unable to pass through the anal pores because they're too big. It eats a lot of other cone jellyfish and can eat regular jellyfish. They probably also eat salps and tunicates, which are some animals we may see here. They're filter feeding animals that look like jellies, but are actually more closely related to fish because they have a spinal cord. This animal here is cone jelly, Burrowi forscali. And as the ROV lights reflect off the cone rails, you'll see like a rainbow pattern. Ooh, it's got a yellowish tinge to the internals, which you normally see on Burrowi matrata, but the structure of the mouth here looks to be forscali. Hopefully we'll have some shots there where we'll be able to see those macrocilia inside the mouth and give that a proper voracious predators. I think that's good. We should have a couple of good frames there. They do eat other tunicates. The Uranthea we saw just before is quite a large animal compared to that Burrowi, but I would put my money on that it can actually eat that Uranthea. They can ingest bits of a jellyfish and using these gelatinous teeth even bite off chunks. The animal that's been predated on will grow back. So he just snacks on an arm and then lets it swim off and grow its arm back, kind of thing. So we're heading here, flying through the marine snow, trying to collect it on our camera like you collect bugs on your windshield. The reason we're flying in this kind of a formation is so that as the marine snow particles come towards the camera, sometime they'll be perfectly in focus. Every particle will be in focus. So we can go to that frame and try and get an ID on what the animal is. Some of the animals can be quite small, like copepods or krill. Other animals larger, like the jellyfish we just saw. The camera on the SuBastian is a 4K video camera, which is 4 times the screen area of your regular high definition video cameras, which will allow us to look at the fine structure of the animals. We're down almost at 160 meters depth now. Would I be correct in assuming that we have come underneath the thermocline here? The thermocline being the depth where the temperature changes rapidly and becomes colder? Often it's such gradients, very sharp gradients in temperature or salinity. You have a lot of marine snow will be sinking down out of the surface waters. As the density of the water suddenly becomes a lot more dense, they kind of float and sit at that depth and provide a high concentration of food. And many deep sea animals will migrate up to the surface at night time to come up to those areas with high food concentration to feed. I see there the CCD profile. Right?
Yeah, we just hit 21 degrees, Dougal.
21 degrees?
Yep, 21 degrees.
Roger that.
Celsius, of course.
Ooh, here we have a little hydrozoan medusa. Pulsing away. 1, 2, 3, 4, 5, 6. Possibly 8 tentacles. The white spot in the middle is its manubrium, its stomach, which hangs down from the central part of the bell. Has that guy got tentacles, or have they been munched off? Looks flattened, wider than the bell is high, which would suggest it's a leptomedusa. in which case we should see the gonads along the radial canals, which will be coming out from that central stomach down along the inner surface of the bell. Hydrozoans, the group that this guy belongs to, is a very, very speciose group. Thousands of reported species. Many, many more than the larger, fairly hard-bodied jellyfish that we see in surface waters around estuaries, for example. Oh yeah, it looks like it's got tentacles still attached. It's probably quite a small animal, I would imagine, which we're trying to chase with a great big hunk of steel hanging from a ship on a thick cable or tether. Oh, there we go. I think I saw some gonads there. Two, three, four, 6, 8. Long filiform tentacles. Okay, nice. Hopefully we'll be able to ID him later. Leptomedusa are a very hard group to be able to identify because they have various structures around the rim of the bell, like the statocysts, which hold the gravity sensing organs, which let the jellyfish tell which way is up and which way is down. There's also sometimes little vestigial tentacles or sensory organs called cordiali, and the numbers and placement of the statocysts, cordiali, tentacle bulbs, tentacles themselves, etc., delineate the different families and genera. So you really have to have a good look at the rim of the bell, which is often torn off when the animals come up in a net. So continuing our slow descent, seeing what other kinds of organisms are living here in the midwater.
You can see the plot of the canyon seafloor that we're currently diving into at the moment on screen.
Right. So steep wall on either side.
Yes, very much so.
We've done some dives off Japan where canyons, even next to each other on the continental shelf, can have very different midwater fauna in them. It'd be interesting to see what we find in here. The canyons act as a channel for food, even at midwater depths, especially if there are shelves as the canyon becomes deeper, because at each of these shelves you often get a jet of near seafloor material coming out into the midwater, So we can see like layers of increased turbidity as some suspended sediments and food, of course, as well, laterally transported or advected out into the midwater. I'm not sure if that will be the case here, but there are certainly a lot of particles in the water, some of them swimming and jumping around, as you can see on your screen there. We have yet to see a kelp or pelagic tunicate, which we saw many of at the Cairns seamount. No, here's something. What's that? Huh. Let's go in for a closer look. It looks to be the nectaphore of a siphonophore Hippopodius, which has become detached from the main colony. So this is the swimming bell of a hydrozoan jellyfish called a siphonophore, Calicophorus siphonophore. And it's been ejected from the main colony, or the main colony has been eaten. And this is just the swimming bell, the nectaphore. It's not pulsing, so it may have been ejected a little while ago and it's already used up the reserves of energy it has within its body. It's a single polyp, a zooid. So where you'd normally have on rocks on the seafloor, little anemone-like jellyfish polyps. Well, this is a polyp that has evolved to propel itself through the water. And it lives in a colony with other such zooids making up the siphonophore colony. Each of them has their own role. Some, their role is to catch food or to eat, others to taste, others to reproduce, others to swim, and others to protect the soft parts of the colony. And this is a zooid that has developed to swim. Thank you for that. Usually with siphonophores, the nectaphores are used to tell species because they are the hardest parts of the colony that survive collection in a plankton net. And we were just able to videotape that zooid from multiple different angles. So we see what it looks like in the X plane, the Y plane, and the Z plane, which will allow us to put a species ID on that animal, which looked like it was Hippopodius hippopodus. When these animals are, the colonies are intact, it looks like a big gelatinous ball with little yellow tentacles hanging down in the middle. And they like to eat ostracods, or little flea shrimp. So probably some of these particles, oh, that looked like maybe an Archimedusa. See if there's any more of them around. A lot of the animals living at this depth are very small and very transparent. So as we're flying through the water, you don't see them until it's almost too late to stop. Oh, there we go. That's some good piloting. It does seem to be an Archimedusa. Very small, very transparent. Looks to have a flat disc-shaped body. With a size like this, it could be Solmaris. And as we look closer, we'll see if it has any stomach pouches or not. The invaginations of the stomach around the bell rim, which can be U-shaped or diamond-shaped, square-shaped, depending on the taxon. And sometimes there are no stomach pouches. And that's also diagnostic for different genera. Looks like it may have stomach pouches there.
Dougal Scott here. Interested in a sample here at all?
I will be, yes. Once we have a top down and a side on in focus shot, would love to have a sample. Oh, and going through the foreground is a radiolarian.
Roger that, we'll line up for the sample.
Great. This group of jellyfish are called the narcomedusae. You can tell a narcomedusae from another jellyfish by the fact that its large, prominent tentacles leave from midway up the external surface of the bell rather than coming from the rim of the bell. You see those tentacles are coming out from around the level of the stomach, and they're often held in front of the animals as it swims through the water. This is because these animals specialize in eating gelatinous prey, and as they swim through the water, they produce a bow wave in front of them when they're swimming. And the large gelatinous prey will sense that bow wave and swim away and escape. So they try and hold the tentacles forward of that bow wave so that they can collide with the prey item before the prey item can sense they're coming. And then they rotate the tentacles around back towards the mouth to ingest them. That's looking good. We can suck him up now, I think. If you would please. 1, 2, 3, 4, 5, 6. So now we're going to use something called a suction sampler or slurp gun. There's a Ketognath in the foreground there also on the right-hand side, an arrow worm. We're going to use a suction sampler, which is kind of like a hoover, a vacuum cleaner, which instead of sucking in air is sucking in water. And we'll position the tube next to the jelly and suck him up as if we're slurping up spaghetti or noodles. Holding our breath. Did that go in? I think maybe we've caught him. So we're panning the camera around now to have a look at the suction sampler canister. And hopefully we'll be able to see a jellyfish inside one of these containers. Suction sampler canister 1 there. Hopefully we will have sucked him in there.
Yeah, Dougal, we can see it in canister 1.
See him? Excellent. So that right now is what we would call, from what I can tell, a cuninid, a narcomedusa, C-U-N-I-N-I-D. However, we found through some genetic work that the Cuninidae is actually a family that contains multiple relatives that are not really very closely related. They're more like cousins rather than brothers and sisters. And hopefully, by collecting animals such as this and snipping off a tentacle or two to do genetic analyses, recording the morphology of the animal with photographs and images, and then putting it in formalin to look at later in the lab, we'll be able to tease apart the taxonomy and see exactly which animals are cousins and which ones are brothers and sisters, and put them in their correct evolutionary phylogenetic framework. One of the things we're doing here with the Narcomedusae is a biogeographic study, trying to make three-dimensional niche models for the different species to see what their habitat preferences are. You have a lot of data from off Japan, but there's very, very little data for the Southern Hemisphere. And here off Australia, the data is particularly interesting because pHs can be quite low. So the water is fairly acidic, and the combination of different temperature, salinity, oxygen, etc. is different to that which we get off Japan. And if we can find the same species, which is genetically and morphologically the same, but they're living here off Australia, we can compare the depths or the temperatures, etc. they live in to see what's more important to them. Are they okay? They can live at any depth as long as the temperature is fine, or do they not care about the temperature and instead only distribute based on the oxygen? And by teasing these factors apart, we can infer what will happen as the global climate changes and the ocean changes as well. If the ocean gets warmer in the surface layers, will these animals still be able to come up to shallow depths to search for food? Or will the warmer temperatures mean they can't get to their food anymore? These are the kinds of questions we hope to be able to answer by doing these video surveys and collecting the animals. We're now at 230 meters depth. Ooh, and another gelatinous animal. Looks like it might be a ctenophore. It's getting close there. Maybe Thalassacalis. That's a arrow worm that just went through the front of the screen. Certainly looks like a comb jelly. Very, very fragile animals. Some of them can have water contents up to 99% of the wet weight. Yes. So this is Thalassacalis inconstans. Just film it for a little bit more, which is a medusoid. Ooh, wow, look at those gonads. It's a comb jelly that looks like a hydrozoan jelly. Wow, those gonads are very interesting. Quite well developed. So those the white patches that look like, if you look from above, they look like eyes in a mask. The common name for this jellyfish in Japan is the mask jelly. And it has two tentacles that hang down inside the bell. It can close the rim of the bell to trap things like krill, for example, if it's caught one, and then hang the tentacles down and try and catch that krill as it's swimming around inside the bell. It's nice. Looks like you might have caught something. Ooh. Are those eggs on the gonads? Could this actually be intacta? There's a ctenophore species that, I think it's Thalassacalis. There's a ctenophore species called intacta, which is has never been sampled successfully, which is why we nicknamed it Intacta. It has gonads that look a lot like this as well. Very nice. Unfortunately, even if we sample this animal, they don't preserve in formalin. So our only choice is to videotape it. That's a nice shot to record its morphology. You can see the mouth there pointing down and to the left, and the long stomach, which has got a couple of things inside it, so it's eaten recently. Young individuals of this species occur in surface waters and can be observed by scuba divers. But this guy's got very developed gonads, and probably as they mature, they move deeper in the water column. Oh man, this could be a different species. It might be an undescribed species. I've never seen gonads like this in Thalassicalis. Wow. Ctenophores are hermaphroditic, or most species are, which means they have both male and female sexual products simultaneously present within the animal, so they can reproduce even if there's only one individual. It's one reason why they became such a problem in the Black Sea and the Mediterranean after being introduced with ballast water. Wow, that's great. Excellent video. Thank you very much. Ooh, give me the heebie-jeebies. Very nice. Okay. Hey, we've got some nice animals here, species level identifications, allowing us to really put our finger on what habitats each of these species prefers. Wonder what's going to come up next. 230 meters, normally we'd be getting krill, whale food, little shrimp-like animals kicking in soon. I wonder if that is also the case here. Another little Narcomedusa there. So if we can count how many tentacles he has. Because the volume of water actually within the bell of these animals is not very large compared to the size of the animals, and they're flat and disc shaped, which is a lot of water resistance as they swim through the water. These guys are probably lie-in-wait predators a lot of the time, until maybe they sense something coming close to them and they do a few pulses forward and catch them with those forward pointing tentacles. It's like a larvation that's been ejected from its house. This is another cuneinid medusa, looks like. Edges of the bell seem a lot whiter than the central mouth part, which would suggest maybe it has stomach pouches. I'm trying to give ideas on these animals from a live video feed sent from Australia. I'm sitting here in Japan, IDing these animals. On the ship, I'm sure they're looking at beautiful 4K video. And I hope to have that back in my lab to have a really good look at these animals later. After we get some good video of this, it would be great if we could also sample him. Very, very shallow if this is a Solmissus. And the water temperature is probably still quite high up here.
Yeah, Dougal, we're at 17.6 Celsius at the moment.
Most of the data we have for shallow living Solmissus off Japan at least puts them at 300 to 400 meters and much colder water temperatures. It'll be interesting to see if this is the same species or something else. And although they look to be very similar, right now they've all been synonymized into a single species. They do seem to live in a range of different habitats and have different numbers of tentacles, for example. So there seem to be morphological differences to tell them apart as well. For this kind of speciation to have happened, they have to have different habitat preferences from other species or be eating different prey. So it's very important to see at the species level what those preferences are. He might have got caught in the wash of the thrusters and we may have lost him. He's very, very small. I'm sure there'll be more around. There's a siphonophore. Floating or swimming polyp colony. Maybe we should just continue heading forward and down and see if we can find another animal that we can come at slowly. I'll try and be a little quicker with my requests for sampling. Next time, we don't get these requests.
Yeah, Dougal, we're just waiting for the vessel to catch up to the SuBastian. Repositioning a bit for a while. Cheers.
Okay. Oh, that looks like Forscalia, the siphonophore. What we call the Christmas tree siphonophore, because of the way the stomachs are arranged around the spiral cyphosome, just like the Christmas lights on a Christmas tree. A few arrowworms or chelignates caught in the wash there. Is the reef we're diving close to now emergent, or is it a submerged reef?
Yes, it's emergent. Ribbon Reef 5 this is. And we spent a bit of last night mapping the outer edge of the reef.
Are we close enough that you can actually see the waves breaking?
Yes, we had a very pleasant evening last night as part of the welcome reception for leg 2. Cruising up as the sun set, watching the breaking waves across the reef. It was nice.
Very nice. This looks like a protist. Single-cell planktonic animal. Does it have a little brown spot in the middle? Trying to see if that's a forearm or a Phaeodarian or what that is. Oh no, maybe neither. What is that? Ah, it's a Calicophron siphonophore maybe. It might be a young Hippopodius colony with a few prismatic nectaphores interlocked together to form a larger spherical Jenga block and yellow tentacles hanging down. Or is it a single, is it two large opposed nectaphores? Kind of hard to make out from where I am. I think we've got it on video though, so we should be able to tell later. I think it was a hippopodid. Physonex siphonophore there. Oh, that looks to be him again. Yeah, I'd say that's a young Hippopodius or Vogtia colony. If the nectaphores are kind of rounded, it's more likely to be Hippopodis. If they're pentagonal, Vogtia. The jury's still out as to whether they should really be the same genus or not. That's nice. So this is probably the colony for that single swimming bell nectaphore that we came across slightly earlier. And that's a radiolarian off to the left as well. Okay, thank you. Quite a lot of particles in the water, lots of good food there. Many planktonic organisms that are floating around. The word plankton refers to anything which floats and cannot swim as fast as the water currents it's exposed to. So they're at the mercy in terms of transport or movement of the water masses that they're entrained in. So I guess if you had a whale with its tail and its fins cut off, it would be plankton. It's not a taxonomic term. Plankton can go from very, very small organisms such as viruses and bacteria, all the way up to siphonophore colonies that are tens of meters long. So a lot of these little white spots we're seeing on the screen are probably planktonic organisms. Small crustaceans maybe. I've seen some single-celled protists, such as radiolarians in the video so far. And the larger animals are the jellies. In the deep sea, where there are not very strong currents or wave action to destroy the bodies of very fragile animals, you can get organisms that have increased their body size by making the body mostly water with very little organic content. And these are the gelatinous zooplankton. And there's a range of different taxa that have become gelatinous, from the regular true jellyfish, the comb jellyfish, mollusks, that's a kind of snail, tunicates like sea squirts that swim around, and arrowworms are also gelatinous, very watery bodies, and usually quite transparent. Here we go, here's something. What that is. Can't see any pulsing. Or maybe a pyrosome? Yes, it looks like a pyrosome. This is a colonial sea squirt. They glow in the dark. If you stimulate one and the oxygen levels are high enough, you'll see flashes of blue light it gives off. The colony is open in one end and cup-shaped. And the little zooids, the individuals that live in that colony, suck in water from the external surface and release the water to the internal cavity. And this flow of water into the internal cavity is ejected out of that open end, allowing the pyrosome to propel itself through the water. So even though it's not opening and closing like a regular jellyfish, and it's just a solid structure, it can still move through the water. And these things can migrate vertically hundreds and hundreds of meters up to the surface to feed, and back down in the deep during the day. He's very, very knobbly, possibly to make it harder to eat him. Doesn't seem to be a shrimp on the external surface. Sometimes there are shrimp that sit there and suck their blood while they're young, till they grow large enough that they can form schools and feed on fish. Seems to be a fairly young pyrosome colony. Hopefully we'll be able to ID him. Thank you. It's our first tunicate for this dive. Tunicates are able to filter very, very small particles out of the water. So in areas where there's not very much food, they can become the dominant kind of larger plankton. This area seems to have quite a lot of food in the water, so maybe that's why the tunicates are not dominant, and we have other gelatinous organisms seem to be dominant. Larvation. Still have to see a definitive krill. Some of the animals, you can tell what they are by their behavior. So even if the camera resolution is not high enough to tell what the animal is based on its shape, sometimes the way it swims or escapes will give it away. Copepods tend to give single jumps, or like 1 or 2 jumps, many times their body length, and then float in the water again, save up their energy, and then jump again. Whereas amphipods and krill will escape swimming continuously without jumps. Krill will occasionally flip away, but amphipods just swim continuously and smoothly. What do we have here? A discarded larvation house? Yes.
Dougal, we've had a question from the crew here about lanternfish. Are there lanternfish present in the GBR?
Yes. Lanternfish have been recorded, depending on the season. It seems like they might do a horizontal migration from other areas and congregate in places such as this canyon. I don't know about this particular canyon, but in canyons such as this, very large spawning aggregations have been reported. So they come from far and wide, find a place that they will get together and have a big party one night, or many nights, I think, to reproduce. So it is possible we will see some myctophids or lanternfish around. Probably a little bit deeper than this. It's probably still a little bit bright, and they'll be wanting to go a bit deeper so they don't stand out so much as their predators, like tuna or swordfish, will be diving down to these depths to eat them. So they want to be down deep enough that they're hard to see in the dim light.
Excellent, Dougal. Thanks for that. Dougal, just a general query about why are we interested in plankton in this particular area from your point of view? We are in the Great Barrier Marine Park.
Well, there's all sorts of reasons why to be interested in plankton from here. In general terms, very little is known about the plankton off Australia. There's been work with continuous plankton recorders that are towed up and down the coast to try and get a handle on what plankton are common in different times of year in different areas, but the sampling is all done at 7 meters depth. There were some surveys done off here over 100 years ago looking at deep water plankton. Very few tows with nets, many new species described, and there really has been very little work on deep water plankton since. So we'll probably have a range of new discoveries we find as we're looking with different tools to these plankton nets that were used, silk nets back in those days. With an ROV, we're able to look at the larger, very fragile plankton, such as the comb jellies or ctenophores and the siphonophores as intact colonies. And the number of data points on large deepwater ctenophores off the Great Barrier Reef before the present Falkor cruise was exactly 1. So we've multiplied by many times the information on the animals down here. This looks like another ctenophore perhaps here.
Yeah, thanks for that, mate. That's great. I guess another element of it is the possible influence of these colder, deeper waters, these Antarctic intermediate waters, we call them.
Yes, certainly the Antarctic intermediate water flowing north. That looks like Thalassocalis again. It's nice. A little bit of video of him. See if the gonads are the same as the last one. Yeah, the Antarctic intermediate water flowing north. What would you say? 700, 800 meters, probably 900 perhaps.
That's right.
Bringing with it. A lot of food. Sorry, take over.
No, the CTD cast we dropped this morning before this dive gave us those depths for those Antarctic waters, around 800. Pretty much bang on where they should have been, we think.
Yep, this is Thalassocalis, definitely. Yeah, so it's very interesting to think of these animals that have been flowing north, trapped in this water mass at those depths. And the environment around them is changing. The food that's being input from the surface waters, for example, has got to be different off the Great Barrier Reef to what it is in the Antarctic front area. So as these animals well, maybe not adapting to different habitats, but they'll probably be changing their distribution depths and other things as they are transplanted into new areas. So it's interesting to compare the distributions of them from further down south to where they are here and what they're doing. And of course, not just the plankton, but all of the benthic animals that are living at those depths are also being supplied food, and that originated down in towards the poles. And if the animals are not as well adapted to these warmer waters, they may not swim as fast, escape as well, and therefore become more important as food as they're moving north. Wow. Looks to be a new feeding filter of a giant larvation, possibly Bathycordius. Looks like it's below the filter there. Looks to still be home. Oh yes. Yeah, it looks like a Bathycordius, the big rounded body, and then a very long, mostly transparent, flattened paddle-like tail. There he is, you can see him there, side on. You can zoom in on that central animal, it would be great. The feeding webs of these animals can be meters in size for the larger houses. This is only the inner filter that we're seeing now. The outer filter has been blown away or discarded, but it can reach 1 meter, 2 meters maybe in diameter. And as they sink down towards the seafloor, they can sink at rates of 1,000 meters a day. And as they sink, they're gathering all the particles that collide with them as they're sinking down. scavenging the particles and providing a very, very rich package of food to the animals on the seafloor when they collide with the seafloor. And there's a smaller with a fluid larvation in front of it. So you can see the two different taxa, the difference in size between the giant larvation and a more regular one. He's still there. Oh yeah, look at that tail, it's huge. And a little salp. Seems to be caught in the web. Recently, another species of Bathycordius was described in the Monterey Bay in the United States. And so the taxonomy of the genus is a lot better than it was a few years ago. So this will help us identify the animal to lower taxonomic level, we hope.
So Dougal, when you collect your samples and we put them in the little bio boxes and put them in the fridge back here on the boat, what are you going to do with them afterwards when you get them back to the labs?
So what we're doing is colleagues on the Falkor there, Jeremy, Marty, other people who are processing the samples, they generally remove them from the sample jar, the sample canister, and try and document them photographically, which is very, very hard because they're very transparent and you can't just shine a light on them and take a photo because you won't see anything. You have to light them from the side or from slightly underneath to get higher contrast against the background. And trying to photograph them to record what their morphology was like. That looks like a prairie siphonophore. And then they'll snip off a couple of tentacles and put them in ethanol in an Eppendorf tube and freeze that inside the ethanol so that we can sequence the tissue later on. And we've been collecting 16S and CO1 sequences, sometimes other genes as well, to barcode the animals so that when other researchers come along later, if we've done the taxonomic work to get a definite species for the animal, a proper identification, and we have the DNA barcode that goes with it, when people haul up little lumps of jelly in their plankton nets that are no longer discernible what kind of animal they are sometimes, they can sequence that jelly and say, it's this species, or no, it's that species, using these DNA barcodes. We also hope to be able to use the DNA barcodes to probe the stomachs of other predators to see what kinds of jellyfish are being eaten by other animals in the system. So this is to be providing a tool for other researchers to be able to use who don't have the capability to be doing surveys with ROVs such as the SuBastian, so that we can use lower cost systems, maybe do more long-term monitoring as well, based on the high quality samples that we're taking here and the DNA barcodes we get from them.
Thanks, Dougal. That was really interesting. Good to hear. We're all on board today.
It's down below 280 meters depth now. Seems to be quite a bit of action as far as smaller plankton go in the water column here. Keeping my eye out for bristlemouth fish, cyclothonae, to see where the first of those turn up. I have yet to see one of those, but they are the most abundant vertebrate on the planet, and also a good source of food for animals living here in the midwater. Not to mention that they are predators upon the copepods, ostracods, and other little plankton that undoubtedly are mixed in with the marine snow particles we're seeing here. Radiolarian. Seem to be noticing more protist plankton during this dive than we had in the first leg. Very large single-celled plankton. Very, very understudied because they're very fragile, don't survive collection in nets very well. Seems to be quite a lot of diversity, and they seem to be more common in oligotrophic areas. It's like a scene from Star Wars, isn't it?
Do we want the theme music as well, mate?
And the rolling credits. Yeah, thing is to keep us going at a nice speed like this so that we, if it looks like we're jumping into hyperspace, we won't be able to identify, oh, a big siphonophore, top right. We wouldn't be able to identify anything, but going nice and slowly, we should be able to. It looks like a praeid, perhaps. Nectophores on the bottom of the animal there. In order to ID it, we'll need to zoom in on the nectophores and have a look at the somatocyst, which is a probably a whitish structure embedded in the swimming bells, and whether it's branching or doesn't actually penetrate the mesoglia, or it does and it's bulbous, the shapes of the somatocyst should give us a good idea about which species it is. These animals, some of them can reach up to 40 meters in length. So very, very large plankton. Each one of those white spots you see along the tail there is a stomach. And each stomach has its own tentacle. So you can see there we have 20 or 30 stomachs. Each one of them can ingest a krill or some other prey organism. Hanging down. Oh, it looks, what do we got here? It's nice. They look to have yellowish pigment. I can't see in the stomachs any food. Where's this? Perhaps. Yeah. That's good. I can't see any spherical somatocysts, so probably not Desmophis. Nectophores are too cylindrical to be a Liliopsis. It's looking very much like a Rosacea. Very very much like a rosacea. Probably the somatocys doesn't actually penetrate the mesoglea, it just runs along the external surface of the nectaphores.
So Dougal, if each of those white bright spots is a stomach, are they connected and then there's the fecal matter comes out at the very end, or how does that all work for the digestion?
They're all, once we have some video of this, can we suck up those nectaphores, please? They're connected via a long tube, which transports nutrients and like the food part, the already digested nutrients throughout the colony. So if one stomach is lucky and gets food, probably it absorbs more than the other zooids does because it's closer to where the digestion is happening, but it shares the food with all of the other zooids. And the excretion is done by after the organism in the stomach has been digested, the stomach opens and just lets what's left drop out. But the actual nutrients are shared through all the zooids. They have to provide enough food to the swimming bells, the nectaphores, to enable the animal to escape if something comes to eat it, or to reposition itself in the water column. I've seen members of this family hanging down at, say, 200 meters or so depth with hundreds and hundreds of stomachs, each of them deploying a tentacle. And then as krill are migrating to the surface to eat at nighttime, they have to pass through this like three-dimensional curtain of death, all these tentacles hanging down, and they get like caught and pulled up and ingested. And then they're eating, those that are left, they're eating in the surface waters, and then on their way back down again, they get eaten again, hit by these large preyed siphonophores. So they're quite voracious predators. Each of the stomachs is smaller than, say, the stomach of a shark or something like that, but they are also extendable. And I've seen a siphonophore where the stomach is only a few centimeters long, which had ingested an eel larva, which was like 20 centimeters long, just manipulating it and folding it over, and the walls of the stomach expanded hugely in order to ingest the animal. So you can't just assume with a jellyfish that because the stomach is this big, this is the size prey it eats, because those stomachs can expand quite large in order to ingest the prey.
Wow, thanks for that. It's cool. Now, would you like to sample this one now? I think you said that.
Yes.
Yeah, we'll go and get it. Thanks, J. Rod.
If we're able to sample it from the nectaphores, the swimming bells, because they're the bits that'll try and escape. The rest of it, some siphonophores have special swimming bells on the tail, the cyphosome, so they can swim even when they've lost the main nectaphores. This guy shouldn't have. They should all just be neutrally buoyant, large gelatinous bracts and things on the cyphosome. And as long as preserved in formalin, I have the nectaphores and part of the cyphosome, the tail. Other little bits that get detached while it's collected can go into ethanol to get a DNA barcode on this animal. We were talking just before about Antarctic intermediate water. This animal looks to be belong to the genus Rosacea, and Rosacea plicata is an Antarctic one of these guys that lives down towards Antarctica in quite large numbers. So the reason we're collecting this is to see whether it's that species, which has found its way up here off the Great Barrier Reef and has come out of the Antarctic intermediate water up into a shallower depth where there's more food. And if it's the same animal, this will prove that they're able to cope with global warming and warmer water temperatures better maybe than some other animals, and allow us to model what will happen in the future as the oceans are warming. Or it could be another species. We won't know until we get it back in the lab. Looks like you got the whole animal there. Awesome.
Bingo. Got landed that one. That's great.
There he is. Yep. Oh, and still all joined together. Very nice.
Perfect effort, eh?
Yeah. Very nice. I must say the pilots on the SuBastian are really top class. It's very, very difficult to follow these animals as they're swimming around in three-dimensional space. The ROV is being pulled around by the tether or moving in three-dimensional space as well. The animals are 95% water or more, and you're trying to suck them up in this canister, and then to catch them where they haven't fallen to pieces. Really, really amazing. "300 meters. This is normally the depth at which the action begins, but we've had quite a bit of action already. Elevation.
Hey Dougal, we're just going to speed up a little bit here, just to get us into a new zone, I guess. Just okay.
Should I put a blindfold on?
Yeah, good.
Very large masses of mucus here and there. Still haven't seen any cyclothonae. Could have been a Fasciact or a dead siphonophore. Oh, it looks to be a siphonophore. Exactly what? So I have two swimming bells and rocket shaped. Looks very small. Oh, is that a jolly old nest? No, it's a Fasciact. It's a Forscalia. Forscalia, maybe Formosa, from the look of that cyphosome. Nice. Forscalia is a little bit different to other siphonophores. Most of the Fasciact siphonophores have nectaphores that are biserially arranged. That means they're in two rows. Forscalia, they're arranged in a spiral. So as the animal swims through the water, they're actually spinning as they swim. Wow, what's that? Oh, maybe a little Haloscera. The tips of the tentacles are whiter than the rest of the tentacle. And the Halocreatidae have rod-like distal tips to their tentacles. That's very nice. It should have wide ribbon-like radial canals. Once we videotape this, I'd like to sample it too, please. These are the sister group to the Narcomedusae. So in order to tease out the correct taxonomy of the Narcomedusae, we need to get more information from their closest cousins, which are the halocreatids, which also have large rounded stomachs rather than thin stomachs hanging down in the bell. And right now, the genus Haloscera, which is what this looks to be, has got a couple of species that need to be resurrected, and it seems quite a few new undescribed species as well. I don't think any have ever been caught off Australia, so it would be nice to see what this is. I'm surprised it hasn't tried to eject its tentacles yet. We don't really know what they eat yet. I've never found any with things in their stomachs, but the fact that the tentacles have these distal ends that are thicker than the proximal ends near the rim of the bell suggests that they do specialize in eating something other than what regular Medusae would eat. but we just don't know what that is. No, he's swimming as hard as he can. He knows we're after him. On an earlier dive on the last leg, we were able to catch the cousin of this guy, another halocreatid medusa called Halotrephes, which had many more radial canals. This guy probably has 8. He might have given us the slip. Oh no, there he is. It's probably extremely small.
Yeah, not quite sure, Dougal.
Roger that. Feel free to see another. It was a good video, though. Must be challenging keeping the ship here in this canyon with breaking waves on a nearby reef as we're dangling down 360 meters depth, chasing centimeter diameter jellyfish.
Yeah, you painted a really interesting picture there, Dougal. The weather is perfect today. Actually no wind and half a metre swell, so it's ideal for this sort of work.
Oh, yes. It's an ocean I only dream about off Japan.
So, Dougal, we're getting close to our waypoint now, so we're going to start coming down faster to deeper depths. And if see anything flies past, we'll do our best to slow down and get it. Yeah.
Very interested in this particular dive, looking for animals in the core of the Antarctic intermediate water at 800 meters or so. The big dinner plate Solmessus jellyfish. There should be a guy with 30-something tentacles down there, which we have yet to sample during this cruise. They're fairly large, so hopefully even if we're going fast, we may be able to stop on one.
We'll keep our eyes peeled. Yep.
Look like a salt chain. So we'll be analyzing this video back in the video lab. Normally on a dive like this, an hour of video would take 4 or 5 hours to analyze, because we have to jog forward a frame, back a frame, until we get the best in focus frame we can of each animal to try and identify it. It's a very long and involved process. Uniform. I think. Is it another Thalassocaris? Looks like it might be. Yeah, I would say so. Quite a lot deep. Yeah, that's another Thalassocaris. Nice. Quite a lot deeper than the others. Excelente. We're also hoping to put together a library of images of these animals to train a machine learning algorithm to help us with video surveys in the future. It's not always possible for a taxonomist to take part in oceanic cruises such as this. I'm very lucky now that I'm being able to take part in this cruise from here in Japan remotely. I could not travel to Australia because of the coronavirus travel restrictions, but I'm able to be sitting here looking at video from 460 meters depth right now and identifying the animals. It won't always be possible for taxonomists to take part in surveys such as this, which is why we are trying to put together DNA barcodes of the animals that we catch, We're also trying to make a library of images to train a machine learning algorithm so that these videos and photographs can be, the animals in them can be identified, at least tentatively, by a computer. And then these vetted images where you're kind of sure which taxonomic group they are can then be sent off for taxonomists for identification. That way the taxonomist does not have to spend 4 hours analyzing every hour of video, but instead they can be sent a few nice in focus, that's a krill, in focus frames to identify an animal. Try and get rid of the bottleneck of species identifications, which is holding back a lot of the science. So it's great to be able to have this 4K video of these live animals in situ. meters depth, where we would normally find the jellyfish Colibanema off Japan. I wonder if he's at the same depth off Australia. Look to be a few more large and meaty bodied plankton around.
Guess this has made all the more easier. Sorry, Dougal. I was just saying this has made all the more easier by the fact we're in the same time zone, more or less.
Yes. I've done surveys where I wake up at 11 p.m. and survey through till 5 a.m. and then catch a couple of hours sleep and then off to work at 8 before. This is much nicer. Only 1 hour difference between Japan and Australia. So right now it's battery on my clock is out.
I would say
perfect.
I agree. It's 11:42 here. Lunchtime.
And in Greenwich, it's 1:42 in the morning. So hello to all our listeners in Britain and Europe. If you're there, good on you for being up at this time.
I've noticed on the chat.
Salps.
Just chatting with the SuBastian pilot here. He's just saying the current is very strong at the moment. It's got it on full thrust, trying to go into it and barely making headway.
Wow.
Yeah, so the speed of the particles is basically the speed of the current at the moment.
Oh. So that puts us on track for the point we're heading to on the seafloor. Is it basically a vertical descent?
Yeah, that's right, Dougal. Basically vertical, but we'll you know, grab stuff for you if we can.
So the current's actually working to our favour then, in that we're getting the particles coming more into the camera than we would be if we had to work down completely vertical.
Good point. Yep, that's right.
That's some big Colobanema. Okay, I can ID that. That's cool. So he's a little bit deeper than off Japan, but still within a depth range I would expect it to be at. This is Colobanema sericium, a Trachymedusa. This guy, the tentacles bioluminesce, and although he didn't eject his tentacles just then, often when a predator comes to eat it, it'll eject several of its tentacles and leave them glowing while it doesn't turn on the lights and swims away, trying to distract the predator with its glowing tentacles. Was that a second one just then?
Yeah, there were two there. So when we slide down to look at that last one, the ROV was moved about 25 metres off course by the current.
Oh, mackerel. That's a strong current.
Yes.
There's another Colobanema. Wow. Is the direction of the current coming down the canyon, out to sea?
That's affirmative. That's heading more or less direct east, I guess, northeast.
Oh, that was either key or you had to actually we'd be able to look at the video later and see if it had filaments coming out of those triangular projections and get an idea on that. That's nice, like swerve so that you collide with the animal. In a current like this, there's not much more you can do. And is that looked like a feeding filter. Target depth is 1,800 meters, correct? I think.
Yep, correct.
Oh boy, a viper fish and a salmonsis.
Tell us, Dougal, if you want a sample. You can still manage that.
Yes, please.
Gotcha.
What's going on with that tentacle? A single, wow. This has got gonad material attached to the stomach pouches. Oh, that was good. Excelente. It looked like it may have been chomped on and was regenerating. Ooh, that's a suggested shrimp, holding its antenna out to either side of its body to sense when a fish or something is coming to eat it. They have little elbow joints halfway up the antennae to allow them to do that. And an oikoplurid house. Okay. What will our next discovery be? Coming down into the Antarctic intermediate water. Ooh, that's another color. That's very big. Is that a color?
Would you like to sample that?
No.
No, keep going. Thank you.
No, okay.
Yeah, current temperature 6.8 Celsius.
Dropped a lot. What depth is the salinity minimum at this station?
Don't recall. Have to get back to you on that. We'll have a quick look.
And another Columbinema. There's quite a few of them around. And another. Columbinema all over the place.
So Dougal, that salinity minimum was 8 to 870 metres, roughly.
870? Awesome.
Yep.
So that will be the centre of our Antarctic intermediate water. Fish. Fish and shrimps definitely are catching the eye more than they were in the shallower depths. Another Columbinema, it looks like. Yes, Columbinema. When they have tentacles that are of more than one length, it means that they have escaped from predators before, since they don't eject all of their tentacles usually when they're attacked. And then they grow back their tentacles. So the shorter ones are the younger tentacles that have been growing back. apart from the Solomythus, one of the other jellyfish that I'm really hoping to see during the present cruise is Pandea rubra, which is a jellyfish that is taller than it is wide, and it's bright red. It looks like a bright red cup. This kind of an attitude. Tentacles are very, very long, up to 20 times the length of the bell itself. And we've caught them in Antarctica. We've caught them off Japan. And they found one or two in Monterey Bay in California. And genetically, the animals off Japan seem to be the same as the ones from the Antarctic. And there's no records of it between Australia and Japan, between the Antarctic and Japan, or the Antarctic and Monterey. So we have no idea where it's living in between. We think it's probably traveling on the Antarctic intermediate water, heading north. But we have no samples at all from anywhere in between, and no records of what depth it might be found at. So that'll be a really good one if we do come across them. There were only something like 10 records in the literature up until 2008 when I published a paper on them and added an extra 60 or so observations. But we still have no records from down this way. Off Japan, sometimes they can be quite common. Oh, that's a lobate ctenophore, having 3 individuals sighted in 2 minutes with the ROV. Have no idea how common they would be down here. And we're just assuming that they must occur here. But with these midwater surveys, just imagine yourself walking out in a field at night with no lights, no moonlight. Even the stars are clouded over with clouds, and you have a single flashlight, and you're walking around on that field looking for grasshoppers. And that's kind of what it's like here in the midwater with us, with our ROV in this massive 3-dimensional volume with what equates to a flashlight, looking at what occurs a couple of meters in front of us and trying to use that information to interpret the ecosystem of the entire midwater system. We know more about the surface of the moon than we do about the ocean's midwaters. Fish, maybe cyclothermia. Like a fluid house. Where are the big jellies? Like a pleurid. Like a pleurid. Like a pleurid. What's this? Ctenophore? Ah, it's the Euramphea with the short 10 terminal filaments. The deep morph that we saw a few dives back. So it wasn't just one isolated animal. Need to have multiple observations because the animals could just be dying and sinking into the deeper waters. But there's another one at roughly the same depth. So it looks like that species does prefer this habitat. Wow, I wonder, it might be a southern hemisphere equivalent of the northern hemisphere Euramphea, which is shallow living, and it's being subducted down with the Antarctic intermediate water to occur at this depth. That would be an interesting line of inquiry to look at. Small Phosphor, I think. There's no Solnicis. Actually very surprised at the lack of gelatinous animals at this depth. Certainly you would be seeing more off the coast of Japan at 850 meters. Tracking Medusa. So maybe they don't survive the trip north. There's something. Sure. Small Trachymedusa, Orcoplurid. So it'll be interesting to see is as we come out of the Antarctic intermediate water, are we going to find more animals below it? Because normally the deeper you go, the fewer animals you find until you get closer to the seafloor. That'll be another thing to look out for. Wow, just above this water mass, there are all those Columbinema, and we enter the Antarctic intermediate water, and there's no large jellyfish to speak of. Wow. Caplurid. But there are fish. Something? No, it's an oik. So how do we get the same species distributed in the northern hemisphere as the southern? Shrimp, Lycoplurid. Doesn't look like it's because of a lack of food, because there are still quite a few particles around. And the fish and shrimp seem to be doing fine. Snork. Siphonophore, maybe? Fish. There's a jelly. Maybe a leptomedusa. Current's a bit strong maybe for small guys. Lycoplurid. This looks like a fish. The way it's swimming looks like a stenopteryx. Yep. Stenopteryx. Cool. He's awfully deep. They've got well-developed swim bladders that don't normally like the pressure. shrimp. Starting to see jellies there. Still no saw misses. Oh, what's that? That's a large larvation house. Could be a Bathycordius, I would say. Can't see the animal inside. Okay. And that's another larvation house, different species. Orchoplurid. So they're still going to be supplying food to the animals living down here. Food for shrimp. Or something big. Looks like a siphonophore tail. But it may be a ghost tail. I cannot see a nectosome. Yeah, it looks to be discarded. Yep. Okay, thank you. Was that a jelly? The mucus particle. Looks to be larvation as well, in origin. Coming up on 990 meters. And the large jellyfish seem to have pretty much left the stadium. There's one. What is it? Oh, Helichrys minimum, maybe? Looks to be. Yes, it is. Can we slurp him up? It's another helicreatid medusa, one of the cousins to the Narcomedusae. This does occur off Antarctica in the deeper waters. And we have several barcodes for the guys off Antarctica already. Has wide ribbon-like radial canals. It should be orange in color, fairly orange in color. A wide, flat, circular stomach. And many projections out on the side of the bell, like pointy, knobbly projections, 8 of them on the rim of the bell. Helicraeus minimum. We think the projections are like a kind of an armor to stop it from being ingested by fish or other predators. Submarine snow. Looks like a discarded larvation house. Yep. Oh, video. Back again. Okay. Squid. Hmm. What is that? What is seen? No. It's quite active. Quite deep. To be able to send a shear clip or some frame grabs to Mike Vecchione at the Smithsonian to try and identify that squid. Larvation house. Looks like another larvation, yes. So definitely a lot of food down here, even though it's below 1,000 meters depth already. So we'll expect to see lots of animals on the sea floor as well. At least compared to areas that maybe do not have a reef or any upwelling happening close by. The current slowed down a bit, perhaps. Seems to be harder to have the particles coming towards the camera now. So we're still keeping our eyes out here, trying to find a dinner plate jellyfish or the red lantern jelly. Would be two that I'd very much like to meet down here at depths where we don't normally get to. There are also, there are also some large Skypher medusae you can sometimes see down at these depths, like the big red jelly, Tiburonia, or Paralia in the northern hemisphere. We would expect to be seeing down around these depths. We have not seen a single one yet, and whether that means that they're not here in the southern hemisphere, or whether we just, on this dive, we just haven't seen them, we can't be sure. But because we have a series of dives here off the Great Barrier Reef over the coming week or two, we should have enough data points to say, well, we're pretty certain this or that do not occur here, which you can never get with only a single dive, or which is often the case. I remember spending over a month on a ship in the Indian Ocean trying to get midwater data, and I ended up getting 5 hours of midwater data for that entire month spent, which is very important data because there was no information about the midwater community of the Indian Ocean collected by an ROV or a submersible. But it is only like a drop in the pan if there's only a single dive. We're lucky enough during this cruise to be able to have multiple transects of the midwater to be able to say, okay, this was not a one-off thing. This species does seem to always occur at these depths or not. Or we have looked through this volume of water and we did not find a single specimen of this or that jellyfish species as well. That will help us characterize to a much better degree the fauna occurring here in the midwater off the east coast of Australia. So larvation. Heading down towards the seafloor at 1,800 meters in an undersea canyon here, coming off the edge of a reef, an exposed reef. We'd expect a lot of organic matter to be channeled down in this canyon providing food for the animals here. We're seeing a lot of pelagic tunicates, lavations, which feed by filtering small particles from the water using their mucoidal feeding filters. It's not clear really whether these have sunk from surface waters or whether they're living here at these depths. We did have some depths in between where there were not very many oikopleurids, lavations. So it could be that the food they are getting is actually being transported laterally down the canyon axis, possibly originating at shallower depths, but being supplied to the animals at these deeper depths. And the lavations might be living here, feeding off that. It's a hypothesis at this time, which careful analysis of the video later should help us to see whether that is indeed the case. When you're here in real time doing a survey, your eyes are looking for big jellies or charismatic fish or squid or something, and it's only when you get back in the video lab and reanalyze the video that you realize, that animal was there, I just wasn't paying attention to it at the time. So we'll have to do a good analysis of this in the video lab later to make sure. Coming up on 1,090 meters depth. We're still something like 700 meters above the bottom, probably. The number of larger gelatinous animals is not very great, it seems. We went down through the Antarctic intermediate water and there were very few. Perhaps a few more after we'd come out of the Antarctic intermediate water, but at the shallower portion of the layer below it. What's that? Ooh. Oh, it's a Paralia, I think. Let's get some video of this guy. If we can't stay on it for very long, just suck it up, I guess. I'm not aware of this having been caught in the southern hemisphere before. That looked like a Pantacogon. Yeah, looks like a Paralia. Certainly does look like a Paralia. Huh. Maybe a different species. If we're able to sample this, it would be great. Yeah, it's a different species. Huh.
Yeah, we're going to have a go at sampling for you, Dougal.
Pimple bulbs are white, not pink, not salmon pink. And there's much fewer. It looks like there might even be two different kinds of tentacles. Awesome. It's our first Skyfer Medusa of the entire cruise. Oh yes. Awesome. Great. There's a very high chance that's going to be a new species. Should be great. It looks to be Paralia, P-O-R-A-L-I-A. at least in that subfamily, Coralline. Wow. Very cool. That'd be great. We'll have a snip of his tentacle and have the whole animal there to look at afterwards. Some of the larger jellies, it's often hard to catch the entire animal. So you have to reconstruct it with what ends up being like a slice of pizza and infer how many tentacles it had originally before you got your pizza slice. But it looks like we got the whole animal there. So very nice. Do not look like the Paralia that we see off Japan or the one that's in the northern Atlantic. I haven't heard of a Paralia in the southern Pacific before, so it will be exciting to see what that is. James Cameron videotaped something similar to that in one of the trenches down this way. I remember I was sent the videos to identify the jellies, and there were some large scyphozoans such as that in the trenches. But apart from, because he only recorded and dived in the actual trench near the sea floor, there's no data for any of these shallower depths like we are in now. It's a very important observation. Coming up on 1,115 meters depth. A lot of particles in the water column. So presumably a lot of food. Oh, there's a little jelly. Trachymedusa from the look of it. So the specimens that have been caught during the present cruise will be housed at a museum in Australia, I believe the Queensland Museum, and at a date when travel restrictions get lifted, hopefully I'll be able to travel there and inspect the animals and work them up for publication. I'm very looking forward to doing. I'm originally from, born in Rockhampton in Queensland, which is the state that we're now surveying off the coast of. Went to university at the University of Queensland. So the Queensland Museum is only a hop, skip and a jump from there. Based now in Japan. Ooh, wow, what's that? Is that a cone jelly? Looks like a cone jelly. Low bait. It's not flapping its lobes, so probably not Bathysceroi. Oh, wait a minute. Huh. What? Maybe a Balanopsid? I guess we'll see when we look at the video later. The comb jellies are one of the most understudied groups of large predators in the deep sea. Oh yes, nice. Very, very fragile animals. Maybe a lampoctade? Perhaps. Okay. I think this could well be the first mid-water survey below 1,000 meters in the entire South Pacific Ocean for a mid-water in situ ROV survey. Anything we find will be new knowledge. Maybe a cyclothermae fish.
The temperature, Dougal, is 4.1 Celsius at this depth.
Roger that. Like being inside a refrigerator.
Spot on.
The temperature in Yokohama, by the way, is about 37 degrees Celsius.
And the humidity?
Pretty high. I have my air conditioner on. Simulating the deep sea. Another comb jelly, perhaps. It'd be great if we can find another Paralia, so I can make sure that's not just a mutant. Especially for the rarer animals, it's very hard to find multiple individuals to do a species description, or it's not based on just one animal. And often the animals are fragile and damaged, so you can't compare the same structures in both individuals, even if you catch two. So it's wonderful with an ROV if you can catch them in really good condition, and more than one, which we've been able to do with Solmissus so far this cruise. The last dive we did, there were very large numbers of siphonophores and almost no ctenophores. That's another ctenophore, isn't it? this dive, we seem to be getting not very many large siphonophores and lots of ctenophores. Oh, what's that? Larvation, maybe. And another ctenophore. Oh, it's a baroid, I think. Yes, it's a baroid, a different species to the one we had shallower, but the same kind of Ziploc bag structure, which can ingest animals several times its own size. Do you know what the maximum depth that the SuBastian can dive to? What's its pressure rating? The GoPro cameras we have on there have a maximum depth of 3,000 meters. We have a one set which is in stereo, 2 cameras in the one housing, which we hope to be using to tell the sizes of the animals that we see and the distance between individuals to try and get quantitative data on the numbers of the animals and the particles. We're using a technique called disparity mapping, where if we can tell what one pixel is, like X, pixel Y, like 1014, X, 612Y on the left camera is the same as yay pixel on the right camera, we can map out the volume in 3-dimensional space. And therefore, when we on the screen using special software, we can click on the same spot on both of the images and measure the distance between 2 spots in the images and hopefully get the sizes of some of these animals. It's very difficult to do with single camera systems. So we use the main camera of the SuBastian, which is a 4K camera, zoom in and focus and get good close-up images of the animals. And the 4K stereo GoPro is widescreen, very wide angle, and this is to get the size of the animals that we are taking with the main SuBastian camera.
So Dougal, we're just having a chat here around our rate of descent and the need to get to the bottom in time to do our bottom transect. And the pilots are having trouble with the currents as we've had all through this dive. I guess just wondering how much further, I know this is the deepest you've been, do you like us to sort of go and give you opportunities for sampling before we get out and go down?
The worldwide data set we've been putting together only goes to 1,200 meters.
Okay.
So as far as comparing to other areas around the world, I think we've done a good job, and we can go to the bottom and see what's living down there, I would say.
Okay. So that's a good call. 1,200 is what we're at right now. So let's push on, folks. Yeah. Everyone's thumbs up around here. Okay, we'll do that.
Thank you very much.
Thanks, Dougal. It was an excellent dive.
Be hanging around if there are any big going past, which will still be recorded with the ProRes video recorder.
Yeah, roger that, Dougal. Thing that flies past, we can sing out ID and we'll keep going. That's good. Thank you.
I know you're aching to see the seafloor.
Yeah, it's the classic, you know, all the high priorities we all have. It's always a case of working that out. And this was
this is great. We have good comparative data set here.
Excellent, excellent.
Anything else that flies by is a bonus. Like the blue.
Just looking at the YouTube chat here from Justin asking about whether there have ever been issues with sharks attacking or chewing on our cable. The answer is no, but sharks have been known to have a go at other scientific instruments that we sometimes behind vessels. Fortunately, very, very infrequent, but can occur. But this cable is, I think, it'll take a decent shark to chew through this one.
I had a shark at 800 meters depth inside a submarine volcano in a dive I did with the manned submersible Shinkai 2000 many years ago that took a liking to the metal sample basket on the sub and grabbed hold and shook us around a bit. Because the submersible is neutrally buoyant, it was actually able to jolt the submarine so that it was like shaking like this. Was quite exciting looking at the porthole at this huge 6-gill shark chomping on our sample basket. Shrimp. Never actually seen a shark chomp on an ROV before. I guess maybe they do, but I've seen them attack an AUV. Because an ROV probably looks pretty scary if you're an animal living down here. A very large number of particles, lots of food. Can't say I noticed any lanternfish or myctophids on our way down. I did see some cyclophonia and I think a viperfish. And then the sternoptics was there as well. The hatchetfish, I think, is the common name. Oikoplurid. Myctophids tend to keep right at the edges of the ROV lights. They don't like to, they can really sense the bright lights and try and keep a bit of a distance. You can often only see the flashes of the reflective scales as they change direction. When they side on, they bright silver and then they turn around and then you can't see them again and then they come back on bright silver. So it's very hard to record them on video and to tell which species is what. Fairly big fish. Possibly a Bathylaca. Haven't seen a single Chrysota or Arctopedema on any of these dives down at these depths. There's a species of halocreatid medusa called Botronema, which looks like a baby's bottle teat, pinkish, which occurs in Antarctica at depths about 1,800 to 2,300 meters. It occurs in about the same depth range off Japan, even though the temperatures, salinities, oxygen, etc. are completely different. And as we are surveying the sea floor at about 1,800 meters, there's a possibility that we'll come across that species. Be interesting to see if it's here, if it's occurring at the same depth or not. Let's say larvation house. It seems like they may have pressure sensors rather than temperature sensors or salinity sensors, and they can actually sense the depth of the water and distribute there, independent of what the light regime is or the temperature regime. Botronema also occurs in the Arctic Ocean, so it's very, very wide-ranging. We don't have enough samples yet to do a proper genetic analysis to see whether they're all the same species or not. But it's a very distinctive medusa if you see it. It's got this big knob coming out the apex of the bell. Oh, wow. That looked to be Vampirotini, the red zipper, recently described from, was the Atlantic? That's the South in the Pacific. I'd seen it before. Would have been the Mariana Islands somewhere. Like a fluid. Ooh, what was that? Like a fluid. Solmissus, the big guy. And the Nypniastes eximia, a pelagic sea cucumber. Also affectionately known as the headless chicken monster. That's a nice deep Solmissus. But we can count the tentacles on that later. Fluid. The altimeter has kicked in at 100 meters above the seafloor, maybe less. Fluid. Probably see increased particle concentration as we get closer and closer to the seafloor. Possibly see more orchifluids and libations as they feed on the resuspended sediments. And more of those swimming sea cucumbers, perhaps. Another swimming sea cucumber. Looks to be another swimming sea cucumber.
About 40 metres to go to the bottom, we think.
Okay. Should be seeing some demersal fish that are up in the water column foraging soon.
Should start to see something shortly, we hope. This is the first time I've ever had visibility at these depths in these canyons.
Can't wait to see what's down there.
Tensions mounting.
Floating up in the distance. Surprised we haven't seen any eel pouts or rat tails or anything yet.
10 metres.
Nichol. Oh, got a reflection from the seafloor now.
Coming into focus. Brilliant. ROV SuBastian has landed at this foot of Okay. Right. Here we are, folks, at 1,803 metres to be precise. The floor of one of the largest submarine canyons on the margin of the Great Barrier Reef, sitting in the Great Barrier Reef Marine Park. We're looking at clearly some seabed, typically quite sandy, perhaps a fair bit of mud, so we'll just move along and get our bearings here. Well, there's some interesting burrows from critters in the sediment. Well, you can actually see one sitting in there, yeah. A lot of biotubation on this seabed here. Black fish, pretty obvious.
Maybe a morit coral.
Uh-huh. Yeah, let's have a look at those. They might be sea cucumbers, Lotharians. There's a lot of really cool markings on the seafloor from the feeding, burrowing traces of the organisms. The circular patterns you can see in the lower right. Picking up, so the ROV is just getting closer to the bottom now. Okay. Let's flip off.
Repellent-emitted jellyfish floating fast above the sea cucumber.
So Dougal, thanks for the dive today. We're going to switch to a sort of a more local commentary from here, just to bring in a couple other folk. So we're going to have to deal with that audio issue, switch you off for the time being. It's all right, but I'm sure you'll keep watching us on Zoom.
Roger that. Thank you very much. Very exciting dive. I wish you all the best for the Adventure thing. I'll be following along on YouTube.
Excellent, mate. Thank you very much, and we'll talk again soon. Okay. Switch mics. So here we are at 1808 metres on the seafloor, 1.8 kilometres below the sea surface, below the ship. And we're going to just look at this locality for its potential to give us a sample of the seabed sediment using the push core mechanism on the SuBastian. So you can just see the claw moving towards all these cores are numbered in sequence so we don't get them mixed up later on. And claw will carefully grab it and pull it out of its housing. Looking at the seafloor here, it's relatively flat and lacking what we would call active bed forms or ripples or sand waves or such. So that's a fairly good indication that the sediment that's deposited here has remained more or less stationary since it was laid down. So this is a good potential site for, what we would call quite an undisturbed record of the sedimentation history. And the reason we want to look at that is that we can take a short core here and use the sediments to identify their composition and potentially work out where they've come from. And in some cases, in fact, also measure the age of the sediment that's been deposited. So we're just carefully pushing that in at the moment.
It's soft.
It's very soft. See a little crab down in there in the lower right.
Nice.
So it seems to have been retained really neatly. That's beautiful. that's textbook. That's.
Yes, it is.
So that's going to preserve really well for us. It looks really quite fine grained, muddy.
I get better each time. So as the mud is kind of solidifying better than it can be stiff.
It'll give us a perfect sample of the actual sea floor. As you can tell at the top of the core, it's immaculate. Hopefully it comes up that way. And because we're in a submarine canyon, there's the potential that some of this sediment has not only come from the ocean, as it were, it's also there's some that might have come from the Great Barrier Reef itself. There we go, dropped in. Well done, guys.
Second one?
Yes, please. We'll take a second core as a duplicate. One thing to always kind of test for when you're sampling in these environments is to get what we might call a replicate or a duplicate sample, just so we understand a little bit more of the variability. One sample can tell you something, but a second sample can tell you a bit more sometimes. So as I was saying, some of these, we're at the very end of this submarine canyon, so we're quite some kilometres from the edge of the Great Barrier Reef proper, but still over time sediments can make their way down the canyon axis, sometimes in quite energetic, what we call turbidity currents or turbidity flows, almost like an underwater landslide. And so they can be preserved as slightly coarser, sandier layers in amongst the muds. There's a lovely zoom-in shot of that core being taken. Wow. Bingo. 2 out of 2. Very quite muddy, light grey muds being seen at the edge of that hole there.
You can get the concave screen wraps.
Great work, Chris. Thank you.
It is nice to see the screen.
Thanks, Adam.
And that's great.
Okay.
Well, a round of applause on board here for that.
So they're saying clients. Yeah.
So the black. Oh, yeah. Okay.
Okay, let's see what we got.
Okay. Those curly things on the seafloor in the lower right, and they're actually quite likely the fecal pellets from infauna, worms or crustacea in the sediment.
We're going to get near them without actually touching it. Oh yeah. Because I wanted to see each one.
See if we can move that.
Alright, he didn't care about us at all.
He didn't want to move. He's asleep.
Doesn't mind it. Alright, it's the first time seeing the ROV. It's like, nah, no big deal.
So what we'll do with these cores, we'll bring them back on board. They'll sit in the ROV canister for the time being. And later on we'll carefully extrude that from those sample holders and likely certainly take a sample just at the very top of the core, which will give us a excellent set of material for the modern day seabed, and then we'll go down in through the layers of the core to see, just to dissect it a bit more and see if there's any variability which might relate to those sort of turbidity currents I was mentioning. That circular feature there is another one of these burrow features which can be formed by either a colony or even a single organism in some cases. Beautifully geometric, aren't they? You know, it's wondrous how sometimes in nature you get these perfect geometries formed. So that'll be an organism burrowing into the seabed in that circular pattern. Sometimes you can, if there are other ways of studying the seabed, to look at the below the seabed structures of those burrows. It's a field of science called ichnology, where you look at the traces of the burrowing organisms, and they often get preserved into the fossil record as well.
So it's a decent size.
Yeah, okay, the two green laser points, what's the distance between those? 10 centimetres. So we're looking at probably 20 to 30 centimetres across.
That's all right. I think for fish do that.
Sometimes fish can make those patterns as well, I'm hearing in the background. So,
I think that's recorded up in Japan, they see that. Generally there's something in the middle that shows.
and these sorts of burrow traces can be very diagnostic or characteristic, if you like, of certain seabed environments and certain depths. So they're a good indicator for us studying the sediment record of
It seems like something.
the sort of depths at which these sediments have been laid down when we find them in the rock record.
Oh, it's another jelly.
There's something flying along here. What have we got?
Deep water jelly. Dougal's watching.
I'm sure.
I'm sure Dougal's watching a deep water jellyfish coming along. That's what that is.
Yeah, it's good size too. I think it's good size.
It's like 10. Well, it's. You got the lasers to the same plane.
Yeah.
What is called?
It's got many tentacles. I think Dougal's been looking for like a 24 tentacled jelly. I don't think.
So we're just lifting off the seabed a bit at the moment. We'll start our transit.
Should we send it up? Jelly?
What does Dougal say?
Okay.
Did you want to sample it? Maybe let's try to take a good picture of it while we wait for Dougal. There's no reason.
It's just decided to leave us. Oh no.
Yeah.
Jar 5 is free.
I'm going to go to audit the manual and then.
Disappeared. Oh, it's still hanging around.
It's a big jelly.
Yeah.
It's not transparent.
I'm going to stay away from it. It's trying to pull away. I want to keep an eye on it in case we want to sample it. We're going to get into a dust storm in a second as well. So.
Yeah.
There we go.
Look at that. Oh, that's stunning. Beautiful colour. Look at
Yeah.
it's just turning around for us.
Yeah, it's awesome. You could zoom in if you want. Why not?
How do I do that?
Yeah, so this is turn. This is in and out. That's up and down.
Okay.
And down is up is down. Left is left,
So
right is
goes
right. We're turning that.
Oh, it's a dial, is it? Okay. Oh, okay.
There you go. Nice. And it's on auto zoom, so it should do the trick. It's a big animal.
I'm not very good with this.
Yeah. So we can go to manual and try to focus this.
Yeah.
Captain manual.
Spin.
So turn this left and right to try to find
This one.
the plane that it's in. It'll turn it a lot.
Okay.
That'll be a big turn.
Okay.
Let me just do
Just getting used to dials here, folks. Day 1, leg 2.
There you go, I got neck.
Okay, understand.
Nice. Perfect. That's it.
That's a money shot.
You're a pro.
Beautiful, isn't it? Jeez.
Yeah, we could probably grab it.
Shall we? We shall sample that.
It's going into
First opportunity at these sort of depths in Australian waters to sample material like this in this way. Dougal Lindsay, our colleague in Japan, will be clapping, I expect, as we do this.
I think you're on a little bit.
If we succeed.
Bucket. Cowboy.
Yeah.
There it is.
Got it.
Little wiggle, just to be sure.
Someone goes, no.
There it is.
Yes, understand the.
Alright.
Right.
Okay, thank you, gentlemen.
Saw it go in, so we're good.
Yeah, we'll proceed, I think,
Alright.
on our transect. So
I'm going to head to waypoint number 1.
Yeah.
Yeah, you have whatever.
So we're just going to track along the seafloor, reminding everyone we're on the floor of the canyon. There's another one of those jellyfish.
Already got your buddy, so
Okay, we've got one of those, so we'll keep going. Leave him alone. So this will continue for a few hundred meters for sure, kind of like this, I expect. And then we're tracking southeast towards the edge of the canyon or the canyon wall, and then we'll take an ascent up the canyon wall up to a depth of 1,400 metres from 1,800, so a total rise of 400 for our transect today. And we really don't know what to expect on this. I mean, this is new territory. We've got ideas, but one of the reasons we've chosen this canyon is because it's one which is connected to the continental shelf of the margin of eastern Australia. It's what we call a shelf-incised canyon. So when you go to its top, it's got a little
bit more
incision into the edge of the shelf, which is very much at the edge of the Great Barrier Reef also.
Oh yeah.
Oh, for the camera. Oh, okay.
Yeah, he's 0.8, so he's really close.
Okay.
So it's an exemplar case of a, what we call a shelving-sized canyon. And over geological time, it's carved its way into the edge of the continental slope, which forms the edge of the Australian continent here in northeast Queensland. And in carving into the edge of the continental slope, it's formed this canyon to great depths, 400 to 500 metres, absolute incision depths, and perhaps greater probably, because there's been some sediment infill over time as well. That's a cool fish. I don't know what it is, but get some great photos of that as we come along.
Figure the side view of it.
If you want.
What are all these feathery things sticking out? See?
On the ground? Anemones, maybe? Let's see.
So that's, he's not sure about us, is he?
Looks like that.
So we're also interested to see what's living in this muddy habitat. It seems like not much, but if we look really close, maybe we could find something. Let's see. Not much, huh?
Welcoming to the commentary, Jeremy Horowitz, colleague on this voyage. Many of you have been following the voyage have heard Jeremy on leg 1. He's still with us on leg 2, which is great.
I'm here till the end.
Having fun.
So all we're seeing are just kind of, I guess, animals making imprints in the mud.
Yep.
So to speak.
Lots of burrows, yeah.
Oh, can we stop here?
Oh, you've got a small coral there.
Zoom out for me for a second.
Yeah.
Something flying along.
Is this nice a fish?
Fish, yeah.
Cool. Let me zoom in on
This thing?
right there. It's
A little.
Oh, picked up the nut of it.
It's all yours.
right through this.
Is it controlling the sounds? Can you use
Back online.
Move out of the way. So the fish, someone called it a halosaur.
Okay.
So
Oh,
Can we pan left for a second? I think I saw something. Yeah, for a second. Oh, it's beyond the arm. That's okay. Yeah, just. I think I saw something. Oh, yeah.
Okay.
So maybe we can carefully approach this and maybe sit in front of it. Let's figure out what it is.
Seems to be glowing.
Oh, yeah.
Bioluminescent.
So we're looking here at a.
Yeah, it's a transparency.
Oh no, it's just the light on it.
Well, we'll wait till we stop. Settle down.
It's geology.
Yeah. Interesting.
Beautiful deep purple colour.
Yeah. Yeah.
So we're just going to try to settle down here. And
That's right, Curtis.
are the lasers on?
Turn them on here, Brett.
Thanks.
It's the old ring, one of those.
Yeah.
It's rough traces, just very what you want. I'd say it's pretty understood.
Yeah.
Isn't it?
Yeah. It looks good. Just zooming into that
Oh, it's like a soft coral.
soft coral. Yeah.
So, I mean, I wouldn't call this a sea pen. Can we get manual focus?
Yeah.
There you go.
Oh, well done. Well done.
So are these are these colonial anemones on a dead sea pen, or is this a colony? You know what I mean?
Right.
I think, but how organized are these anemone-looking things? Now they look random, so I think they're just on a dead stalk.
Okay.
So these are, I think, colonial anemones.
Very strange. I'm going to say soft coral straight up, but they're very. Just looks really different.
It just looks weird. I mean, if they were, yeah, those are not tentacles and polyps on a soft coral. Yeah, these are
They wouldn't have as much
anemones.
of a body, would they, if they were just the
Right.
heads of it.
Yeah, and you could see this, like it's not organized the way that there aren't rows of polyps. So we're not gonna touch this, but it's cool, interesting to see. So they're utilizing the minimal substrate to attach to.
So that little shrimp there. No red.
These white things, these could be pteropods. That's an amphipod, I think.
Yeah.
Amphipod is vertically, yeah. So that's an amphipod. These are fecal pellets.
Yep. Yep.
The little white things, they look like plankton. Foraminx.
Bivalve?
Yeah, it looks like some kind of pteropod, perhaps.
Yeah.
You know, a swimming gastropod, or pelagic gastropod. These look like pteropod pieces.
Yeah, right. When we were dredging, every dredge had many of them.
Yeah, and they live in the water column, they die, they just drift out.
Yeah. Yeah.
Lots of different shapes, you've got star types. Yeah,
That looks
these are
like-
pteropods, yeah.
Yeah, getting in this close is really showing us how undisturbed the seafloor is. All these fine scale features to be preserved.
Yeah.
A big pile of
brilliant.
fecal pellets that
Yeah.
haven't been completely broken up.
Yeah.
Who knows how old they are.
If we look straight out ahead, they're flat.
There's a landscape.
Oh, there's something up there.
Something in the distance. Is that on?
It's good to move on.
Let's move on to that. Yeah, thank you. We can
Yeah.
Head on.
Any,
Yep.
any mound will be really interesting because that might provide substrate for.
Exactly. Could be a rock.
That'd be really interesting.
There's a rock or something right in front of us.
I love this mud stuff. You know, if you're a diver, muck diving, you see all this life at PNG, and you know, in here there's all this marine life, and it's undisturbed, so you're getting that accumulated life that just builds up and builds up.
Oh, we got coral.
Goes away.
Oh, wow.
All right. Looks like.
Cool here.
Oh, maybe not. It looks like a mud. It's not like a mud.
It's a block. That's a block.
Bit of a debris.
I think it's part of it. Rob and I are looking at this. It looks like a debris deposit potentially. A block of clearly
Oh, there's
very fine-grained.
what's on there.
It's covered in pelagic mud.
Oh, are these coniferous sponges?
Something living in it.
What do you think, Rob? You think these look like what we found?
Yeah, they could be. This
I think-
is what America's after.
I think so. Let's we settle down and look. We're interested to look at up here. Those things.
So I'll come a little bit to my left.
I'll zoom out for you.
No, you're fine. I'm just right next to the camera.
Oh, I hope these are these coniferous sponges. Oh, and there's glass sponge.
This is one of these
Tulip. It looks like beautiful.
We collected what Mary started.
Oh, okay. Mary. Right, yeah, Mary.
These are coniferous sponge.
So this is a glass sponge, and we collected one of these on the first leg.
Except vanilla, this is fully silica. If there's something growing on it,
Probably rhizomes and
or those little anemones, you know, they attach into the stalk.
yeah. And you see, it has a long stalk. It might need to have, stay off the seafloor and continue to grow upwards to survive.
It might clog with sediment, perhaps. That's it's evolved to do that. More bumps out of that.
Yeah, I think
So I mean, if these are blocks, over time they're just getting
exactly. Yeah.
You get this when you see all of the bites of the burrowing.
Yeah, on it.
Bitumation,
Yeah.
So
This is good.
Okay, let's see what we got here on this. Maybe, right? Let's see.
these almost look like follicle tubes. I know they're just hanging there.
Around,
Yeah.
but it's not too bad.
If we could just zoom, no. No, they look like, Can I get manual focus? I'll try to.
That's manual.
It's on manual. I think I'm in the backgrounds.
I can try and spin around to the side and have it in the forefront more.
Let's try to, on automatic, it should focus on this, right?
Let me just kick it back in the
Yeah.
There it goes.
Okay, so these are,
Look thin. No, there's a series of tentacles around them.
yeah.
What the hell is that?
Oh, wow.
Is this what America's are?
I mean, these are not sponges, right? These look like, what are these, like hydroids? Okay. It's
I'm looking
interesting.
at a rock. Worm tubes coming through it as well.
That's really interesting.
It's beautiful.
I wonder if we should we take one to? It's amazing. It's so weird.
I can even guess. I mean, it's sponge-like. I mean, it's Nigerian-like. It's got.
But it's radial. It's radial, like up and down.
Do we
It's,
have forams that look like the benthic forams that have?
It is.
But it's got a slope.
should we try to go to the other side? Or that one looks like it's facing forwards. Let's look at the.
There's a
There's a
lot.
little colony.
They're all
Yeah.
Oh yeah. Maybe someone.
Right now I'm right here. They're all right there, so I can swing around that section right there.
Yeah. Okay. Why not? Oh, is it going to kick up a lot of sand?
I'll try not to.
I'm trying to get a sense of the lithology, but it's a bit hard.
It's yeah.
It looks stiff.
Yeah, it looks like a mudstone, but that's probably because it's covered in.
All right, we'll zoom out for a second.
I just find the safety.
Yeah.
Oh, there's a bit of down on it. There's some fresh face, I think. Yeah.
Weird. We should get
Like giant hydroids collected on dry dive.
hydroid.
Someone's saying hydroids.
Yeah, I mean, I don't know what else I would call it. I guess a carnivorous sponge. I don't think so. I'm not sure. I mean, I guess we could kind of see if it's moving. Are they spicules or more like tentacles?
So you're going to try and catch one, get one, or is the problem is going around the other way? You've got the current behind you.
Yeah, it would be hard. It'd be like a
Hopefully we're
dandelion.
getting some fresh face in this block, Rob,
Sure.
as we come around. It looks a bit darker.
This could easily be a temporary block or a
Yeah.
big chunk of mud that slid down
Sweet,
the
yeah,
side.
yeah.
Look at these things. They're
It looks quite different to your background.
Absolutely,
surface.
absolutely.
But yeah, look
The
at
angle
that.
bit
It's
of-
fringy.
yeah, much better. Thanks. So we'll try to get a zoom in.
Yes, manual.
Thanks. So that means, I mean, that's the face of it. On both sides, it look the same.
Yeah. Sitting there waving in the breeze.
It has almost like a forearm look about it. Like in
Yeah.
you know, it's got that flattened.
Should we get a?
Big forearms, I think of them as completely.
Yeah, exactly.
They have these little pseudopods that they put out, like little slimy pseudopods. Like
Yeah.
Forehand, this is how they feed, and then they bring the pseudopods back into their test to, for the organisms. It's an organism inside. I've never heard of one with a stall.
Should we try getting a sediment sample with one of those?
Yeah, let's take that then if we can try and...
Is a scoop gonna work?
I reckon so.
Yeah.
This would be soft.
Yeah, if we can get some of
So if we can get-
the rock block. Some left.
We have
The scoop? Yeah.
try to like just
Try and
take it
block.
Yeah.
The only thing with the scoop is once we scoop it, do you want to leave it in the scoop and try to protect it in that bobos? Or
Why don't we put it in a?
we can also
Suctioning it would probably rip apart the. And I don't want it to mix with the sediment too much. Can we put it in one of the A? Or is it taking space of a very important rock?
No.
Not yet, because we have to get to the rocks.
All right.
we also have
How about taking a small scoop and just putting it in BioBox 2, and we'll know it's there because that's the only sediment we'll put in there.
Yeah, thank you.
I'm trying to grab 2 of those while you're at it, if possible.
See how soft that rock
So
is.
I'm gonna put that down.
Might be 2 for 1.
These carnivorous sponges, the small ones that are next to them.
Alright, stand by. I'm gonna take the camera, guys.
They look like trees. They have yeah, it's not what those look like.
That face there.
Oh yeah.
That's like sand. It's quite a...
It's very burrowed, isn't it?
Yeah.
Oh, this is exciting. I'm excited to take a look at and image it.
Watch out some more.
I like
Is that
just
something
things that we just, you go through your whole mental undergrad phylum list and go,
No,
I don't know what that is.
no idea. Yeah, it looks like
Hey, Jeremy?
Is
I'm
it?
not sure if this will work, but we also
Yeah.
have one quiver on the starboard rail. We could take a snip of it with the coral cutters and try to.
what did the coral cutters look like? You mean the arm,
Um,
or is it a small?
yeah, so you see right here, we've got these two blades and then the little soft jaws.
So
So if we come in very gently, just take a little snip, plop, and then into the coral, into the quiver. And the quiver is just one container with like a rubber lid and just gem lid on it.
okay.
Yeah, try that. See how that.
Yes.
Oh yeah, that's not
That could be a zoid, I guess.
The ones on the rock now are
One
hydroids.
second. It's probably not the most.
Okay.
Safest.
Oh, that's James.
Looks like it anyway.
James works with two.
Oh, okay.
That's all we have.
Okay. Yeah, he lives in maybe. He thinks hydroids.
Maybe.
Yeah,
Yeah,
hydrant's a
see
good
what
guess.
Everyone says maybe.
He's a zoanthid expert. Like
Okay.
he's world zoanthid expert.
All right. Okay.
Yeah, Dougal. Yeah. So James and they work together. James works in the Ryukyus in Japan.
By the way, keeps getting it.
You're going to be doing it all to get this done.
Fortune or
You're going to be doing it all to get all the way out.
Alright. Fortune in.
Tomorrow if we have to.
That's not cooperative.
Yeah, I think actually if the hydroids produce geoids, okay. So the money's on hydroids, eh?
It's good, guys.
How bloody is that?
It's a muckduck.
You know what I'm talking about, right?
Yeah, frogfish and wonderpuss.
Yeah.
The mud block thing.
Do you have a particular one that you want or any other?
Just the corner, so you can get a few of them. And we'll try to preserve one nicely. Preserve them all.
If you get all that with the core cutter.
Yeah, I'm off to the side.
Alright, so what he wants, he wants
Yeah.
this side right here.
Yeah,
They're all,
yeah.
all of
Grab.
that.
Scoop it. Just kind of
So-
scoop up and see I get
Yeah,
it all.
there's like
Yeah.
6, 7 of them right there. So
It's
just
probably going to be a little muddy if that's all right.
Yeah, that's okay.
That's good.
Yeah.
Because it's
We
a
all-
boulder. We think it's a debris boulder, so
Getting some
we
of that.
get
helpful.
some of that as well.
Cool. And I guess these are others.
Could be.
Yeah,
There was another
the
one.
debris flow.
Bumps in the back.
Look at that coming into add to the shot.
So
They're tiny, aren't they?
current makes them face into it, and then the tentacles come out.
It's mythology.
Oh, good scooping. Look how soft it is.
You don't want to turn away a bit.
Oh yeah, we've got a few in there.
Oh, did they?
Yeah, there's two in there. I can see there's a movement there, and there's another
How
one.
about I just
further.
drop these in like 1A, and then partition, and then go back and get another one?
You can put it 1A, and then look, we can throw a rock on top. We're gonna know what's what.
Yeah, sure. It's a mud block, I think. So,
It's a bit stiff.
yeah.
But you could squish it between your hands.
Yeah.
It looks a bit more stiff than the surrounding.
How far was this over there?
What?
How far was this going?
How far out?
Is that orange?
How far does it go?
Is it as far as it's going?
I think so, no. I can go one.
Yeah, I think you're going.
Yeah. I'll just,
Come on.
stand by.
Real wide experience right now.
4D. Box one.
Yeah. Yeah.
Did you just break the chair? I like you broke the chair. Alright, so right now let's have block on. He's gonna go back in and get another scoop.
Yeah, I've got it.
Top right part of this little mud wall.
Fall apart a bit.
Yeah, we got a couple on this one. Let's go.
Yeah, that's great. That'll,
You gentlemen, that's it. Tell him that.
nice work.
All right.
And you want one more?
Perfect. Nah, I mean, one time.
Yeah, thank you.
Yes.
Thank you. We're happy. We can move on.
You want to get a couple more though?
Nah, I think
No, we'll keep going. We've got a long way to go.
I didn't even pay attention to what you said. So.
I'll just show you this graph on HD2 right when we got to the bottom, so we killed him.
Actually, two of them. Is that starboard or?
It's this guy here. It's that nice one.
Oh.
You could try it again. Fire it back up. For some percent where it handles a little bit loose, so it twists a little bit. That's good. What do you do this guy after? You can just drop it in the box. This guy's open then. Just mark it by output. And you got a 678 back from the
Not a happy girl.
just take a look at it. What I've been doing is current. You put it. Current's been going from south to north. So your sail is like this.
Yeah.
So I'm moving the ship kind of ahead. So I'm just taking a big piece left.
Sweet. So I'm gonna put that up.
There you guys go. Have fun.
Thank you, sir.
All right.
That was unexpected. But no,
Yeah, weird. Interesting. I mean,
this far from the canyon. It's a long fence.
And then we'll put that. Are we going to use the scoop again, or just pick it up?
we could put it away. I mean, you could, yeah, I think so.
Just in case.
Of course, we're going to find something to scoop in a minute, but you know, that's how things go.
Yeah,
That's how
yeah.
far we are.
We'll probably see more of them.
Alright, Scott. Why was that so interesting?
Well, we've just seen a block of probably mudstone on the seafloor, which is a good distance, hundreds and hundreds of metres from the canyon wall. Probably come from the canyon side itself as part of a big landslide reef line. Early days yet, we'll see what else we can see along this track, but we start to see more of this evidence. It's all part of that geological history, this margin evolving and incising from the canyons and so forth. Thanks.
Hey, Jason.
You're natural.
You might be right. It might be a carnivorous sponge. Honestly, it's really hard to say. But what we'll do is when we bring it up to the lab, we'll be able to see how rigid it is, and we'll preserve it and send it to Merrick Hickens at the Queensland Museum, who will let us know, will tell us what it is we're looking at. If it's not a carnivorous sponge, potentially it's a hydroid. But we have experts who were able to contact all these different groups, so we'll get to the bottom of it. And we'll let you know when we figure out what it is. If we could just hover over these little mountains, we don't need to stop at them, but just kind of see if we're seeing the same.
Yeah, that's right. These could well be more of these blocks which have been buried by mud in over time. We'll see.
What's that?
Biota, for sure.
Two little things in there.
Zoom in.
This is.
Yeah.
I'm still learning to drive this. Sorry.
Yeah. Oh, it's is that? Oh, a Ribbon Star.
Brittle star going into its burrow.
Cool.
Kind of beautiful.
The other way.
Thank you. Oops, too fast.
Okay.
Excuse me, folks. I'm just learning to drive. Got my
first day I did it, my
L plates on today.
the first time I did that, my partner said she was getting her dizzy. So.
Be more gentle now.
There's no zooming while we're on the transit, Scott.
Are we doing the transit now?
Not yet.
Oh, nice.
Good.
Yeah. Okay.
Okay, got a good shot of that guy. Move on.
You want to look at things to the right?
That was an optical. Did you have a coral off to the right?
Yeah, we're heading. There was some sort of.
Yeah.
Looks like it got buried. More of these anemones, these colonial anemones that seem to take over dead. Whatever this is, this could be a glass sponge stalk.
Get a little closer to it, and we'll do a white balance on it.
Okay.
So it looks kind of green.
Yeah. So if you guys
Okay, cool.
if you remember on leg 1, we collected a tulip glass sponge. So, and we did see one earlier.
Real quick, sir.
Yep.
So do you think this is? Nope. So I think this is the stem of one of those tulip sponge, glass
They've been
sponges.
colonised by anemones.
Right? It looks similar.
Yeah. It looks old. Like the stem looks old.
Yeah.
I'm going to put brittle star behind it.
So if you want to practice your focusing skills, you could try to focus in on these colonial anemones.
This guy?
There. Yeah, that's it.
Yeah.
It's on autofocus and beautiful. And you could zoom in. And yeah, these are certainly colonial anemones on the dead stuff.
Come on. Well, that's brittle stars moving down there. Will that focus at this distance?
Zoantharians. Yeah, it takes a lot of school.
Yeah. I'm not sure whether it's coming now. Great. If you turn too fast, does it upset it or?
Nah, you go as fast.
Oh, okay.
There you go.
Yeah, thank you.
So
That's right.
yeah.
It'd be zoanthus, yeah. And James Ryan would know. He's the world expert in zoanthids.
Sorry. There we go. It's a bit better. As well as the brittle star.
Yeah.
Okay.
It's on auto.
Yeah. Well done. I will pay good Japanese beer for that assessment. Well, can we? We've got a permit that allows us to do this.
Yeah.
We have. Yeah, we
Yeah,
can
we
do
do.
inverts.
Yeah.
Can we do a pluck or a clip? Maybe
We could cut an end off the stalk. I mean, it's a glass sponge. We could snap a piece. if we want.
Yeah, we have a permit that allows this.
Since we're here,
All right.
Yeah.
Let's We'll just.
there we go, mate. Just let me watch you. Okay.
Is it on manual? So.
Yes. Let me reset mine.
Yeah. Okay.
Now try.
James is at the University of
How do I?
Seems like it's not doing anything. So,
Yeah.
Okay. Let me throw it on auto, and then I'll
Yeah.
see if I can manually.
Yeah, I bet you can.
Just in case.
He's American, but he's been living in Japan.
Jason said he wants some expensive Japanese beer.
Mm-hmm. Okay, I think we can do a trade.
Yeah, well deserved.
That's a pretty easy, good trade.
I
I don't mind Japanese beer.
Yeah, that will take like 6 inches off the end.
He's blue. 5? That's fine.
Yeah.
It's one of
So 6 after, obviously.
Yeah.
You know. And also have to figure out which is the end. Seems to be caught up with ends. Oh, there it is. Nice. Okay.
So you want to see where the end is?
That's
I think that's the.
I guess take your claw and kind of hit aside.
Give it a little
to see.
tip that.
That looks like the base.
It does look, yeah,
So,
it's just bent over there.
yeah, so cut like there.
Got that Benjamin?
Yep.
Yeah.
And her. Where would you like to put this one?
Oh, you can put it in Biobox B. Two. Sorry.
You want to throw it in 4/4? If we have one on.
But that means only one goes in there, right? It's not worthy.
It's not really.
I like it, Jared. We'll grab it, Ben.
Salama. Oh, it's definitely glass sponge stock.
Yeah.
Come down some. Come down, come down, come down. Yeah, enough?
Sure. That's even more than enough, that's fine. Yeah.
I wouldn't mind having a chat. Let's go live. I wouldn't mind.
Oh yeah.
Is that all right?
Yeah.
So how do you do it?
Just speak.
Just speak.
And then if you want to control,
What about
zoom in, zoom out.
what about muting?
Box 2 coming open. Close you got there, Ben?
So you don't use that?
Not anymore, no. Only when we have someone calling in because there's feedback.
Ah, right.
And the sound is way better. Like this, the sound is better for everyone else.
Yeah.
So you can see how the zoanthids suck their tentacles in, now that they're disturbed.
Yeah. Well, this definitely shows the zoanthids. If that's what they are, they're alive.
Biobox 2.
You can see that all the tentacles have now withdrawn, but we're pretty sure that it was on the stalk of a hexactinellid sponge. So what we've been seeing is these sponges have long siliceous stalks. Quite strange, really. When you pull them out of the seafloor, they look just like a fibre optic cable. They're almost entirely intact, twisted around like the strands of a rope. And clearly these zoanthids have colonised it and just worked, completely covered it. I think that's when we first saw it, we were quite confused by whether it was a soft coral or not, but just the whole combination of the polyps just look utterly weird. So I think we've now nailed it that these are zoanthids. I think we've got James Reimer from the Ryukyus in Japan watching. Thank you, James, giving us the hint that that's probably what they were, the zoanthids. And James is a zoanthid specialist. So great to have you watching, James. In fact, if I recall, you and I were on an expedition together out in the Great Barrier Reef, I think around 2011, when Dougal Lindsay, our plankton expert, came down from JAMSTEC from Japan, came to the Great Barrier Reef hunting the elusive plankton. He brought his Picasso machine, which is a very quite amazing ROV. It's quite different to the SuBastian ROV that we have here, but we managed to get Dougal's Picasso ROV down on the side of Osprey Reef, which is not far from where we are now. Anyway, we're going to continue our transect now. The aim is to head up the sidewall of this canyon. As you guessed, this is a fairly flat part of it. This is the floor of the canyon, and we've got a decent looking fish in front of us. So reaching out to our, and thank you, Dougal, yes, it was the Picasso, any of our fish army out there, can they put a name, can anyone ID this fish? Has interesting looking, almost like a lure sitting on top of its head there. Bit difficult to see from behind. Fairly slow moving. Doesn't seem to be too disturbed by us. It's got really large eyes.
That's right, yeah, 3.
Here we go. Like the top of its pectoral fin looks like it's moved forward, almost like that, like the lure of an anglerfish. Moored cod, thank you. Thanks. Yeah, it's from Dougal. Yeah, okay, so I'm a bit of a fan from the Lebensspüren Club. This is German for life traces, and if you look beyond all the floating plankton and the fish, there's this utterly beautiful moonscape of life traces, and these are all the imprints and burrows and mounds left by countless crawling animals. And you know, you've just got to look a little closer, you can see all the patterns there. But one of the things this tells us is that the seafloor here is very undisturbed, and so we might be looking at the life traces of animals that have passed through here over many years. You can see sort of fresh tracks down there, but you can see these weird round imprints. Now, we really don't know what's caused these, but you can actually look closer and it looks like this is a series of burrows. So probably some infaunal animal, perhaps some kind of polychaete or some kind of worm. There's lots of worms and things. Oh, we've got a lovely shrimp coming into view now. This deep crimson red colour. And you'll notice the antennae sort of streak behind, and I just recall one of Dougal's chats earlier. They have that streaking behind, and if they're disturbed or a prey animal comes within close distance, it's almost like a little radar. You know, it immediately detects any movement, and then they can then dart away. They're clearly quite good swimmers. Lots more shrimp. So generally the mobile animals we've seen down here are well, on the seafloor. We've seen brittle stars, we've seen pencil urchins. There's quite a lot of these drifting jellies, these deep crimson dark jellies. We've collected a sample of them earlier. We've got eels.
Right there, at the seafloor. Come up 2 meters.
Awesome.
So we're not allowed to swim the camera.
Oh, that's quite beautiful. Another bit of plankton drifting past here. Oh yeah, it's a Medusa. Dougal, are you watching? What have you got there? These beautiful colours here. So the almost dark purple tentacles with a yellow inside bell with striped purples around the outside of the umbrella.
That's really
Cressida milsae, I think. If I read that correct. Yeah, so this is Dr. Robin Beeman. I'm on the audio at the moment. It's fairly crowded here in the control room. I have to remind everyone this is the deepest camera's ever been on this part of the Great Barrier Reef. So this is really quite unique, what we're doing now. Yeah, so this looks like a feather star of some kind, perhaps a comatulid feather star. These are free swimming. They can actually move around. We have other stalked feather stars. We've seen them on previous dives where they're attached to rocks, and they're usually on a much longer stalk, but this one's a bit hard to see. Looks like it's sitting up from the seafloor. We got Basingid. So this, if that's, yep, Basingid, this is a sea star, not a feather star. So just stand corrected there. And in times, some of the previous ROV dives we've had in that Coral Sea, we've come across rocks absolutely covered with these. Oh yeah, that's a nice view now. Yeah. No, someone's saying crinoid. Yeah, I'm thinking it's more of a crinoid. It's feather star. It's much easier to see now that it's in focus. And you can actually see the mouth on the upper surface. This is a comatulid feather star, so it's free swimming. They're quite interesting in that in places we've seen them in dense numbers, if they're disturbed, they can actually swim with this beautiful floating armed sort of movement. I did a voyage back in Antarctica several years ago where there were lots and lots of these, and you know, as the camera was drifting over the seafloor, they would all sort of push themselves up into the water column, and they just drift with this beautiful movement. But you can see these are the legs. They're much shorter, they're only a couple of centimetres long, and you know, they can actually attach to things. And on the shallow parts of the Great Barrier Reef around coral reefs, that they can be in quite large numbers. You know, obviously different species to what we're seeing here in the deeper ocean. But on the shallower coral reefs, you know, they can fringe. They just hold themselves up in the water column with their arms out. And you know, as a scuba diver, it's quite beautiful to see a lot of them together. You know, it does look like this great garden of arms. And in some other places we've been on the Great Barrier Reef, we've come across such large numbers of these feather stars that they look almost like seagrass. You know, it's just literally thousands and thousands of them together with their arms waving in the water column. Okay, so we're just passing another feather star in the centre view.
It's on auto, so it's not taking that live marker. It's just
Title light. Not what I'm saying.
You can once you get your eye into the patterns on the seafloor, some of them make perfect sense. So we just went over one with a five-armed trace on the seafloor, clearly made by an echinoderm, which have this pentaradial symmetry, you know, variations of five, and so that was five-armed, could very likely be you know, a sea star. And we have seen them where they actually push themselves up off the seafloor, like the body part, and the arms are actually pushed down onto the seafloor. And when they move, they leave this quite distinct five imprint on the mud surface. Is that real? Okay, looks like we've got some kind of change in slope coming up ahead of us. What we're aiming for is the sidewalls of this canyon. So we've been down at around 1,800 metres, and we're basically heading laterally across the base of the canyon with the idea to start going up the sidewalls. Now the sidewalls are literally just the side of a valley. Think of this as one big broad U-shaped valley. The base of this valley itself is about 1 kilometre across. That's the flatter part of it. And we're heading to the sidewall, so we should actually start seeing a change in the gradient of the slope, and that will show us as we're starting to go up the higher gradient sides of this canyon. Oh, here we have a nice inset view. So you can see that that long downward swim. That was all Dougal's dive, as we call it, and we're just at the base of that the valley side. So this is the sidewall of the canyon. The canyon itself, we're just calling Ribbon Reef, the canyon in front of Ribbon Reef 5, because that's the name of the closest coral reef that's just west of us. It's about 8 kilometres west of us now. And you can see how steep this is. In reality, it's probably around about 25 degrees gradient. So if you're in a car, you could probably drive down it, but it'd be quite a steep drive down. Where we are in the valley, though, it's completely flat. Okay, so looks like we're going over some kind of holothurian, a sea cucumber. Now, this animal on the right, this does look like a Brisingid sea star. They typically have this very sort of pink-red colour, and it's interesting how it's got the tips of its arms coiled up. Now, just keeping your eye out, there's a brittle star over on the right. Have a look, some of these burrows, right? Some of these burrows we've seen before. Now, I am thinking that this could be one of these giant isopod burrows, and I would love to see a giant isopod, because we have seen them before. And, you know, when we're out on the coral sea, we're on this kind of environment. It's very muddy, sort of a stiff mud. And what you get are these holes, and here's one coming up now. They're flat floored, and they've got a dome shape. So, you know, fingers crossed we're looking at giant isopod burrows here.
How deep would they be? Should they be very deep or shallow?
Well, when we actually found them with isopods inside, the tail of the isopod was sticking out. So, but there were always a couple of other burrows around. So I don't know whether they're actually joined internally, like underground, or if they're just single burrows. But this is really a classic shape of one of these giant deep sea isopods. Like they're big. They're like a giant pill bug. They're about as long as my forearm, about as fat. And they have this, you know, the bottom of them is perfectly flat, and they have this dome carapace above them. So that, you know, they're a crustacean. Okay, this is a pteropod, right? So this is a pelagic gastropod. So it's a member of the mollusk family. In fact, actually, if you look to the left of it, there's a couple of others here, scaphopods, which are, what's the common name for them? Tusk shell. So we've got a number of different kinds of pteropods here. Bear in mind, we're really zoomed in. It's quite fine scale here. So those tusk shells, the scaphopods, may be only a few centimetres long, but they look exactly like a tusk. They're white. They're slightly hollow. But when they're living, the animal is floating around in the upper water column. And they're a pelagic animal, and when they die, they drift down as part of this debris, this rain of debris that we saw through earlier through Dougal's dive. So if we were actually to scoop up some of this mud, you know, it's quite slimy, but in amongst it are all this sort of coarse material, and we've got, you know, the remains of pelagic shells, you know, these pteropods, which have all these different shapes. So there's another, looks like another there's another tusk shell there. Something orange, I can't make out what it is. You know, but clearly, you know, there's a lot of material here that's drifted down from above, and it's making up the sediments that are here. So the sediments come from two sources. They're the rain from above. This is the pelagic rain that we've seen through our midwater column dive. But there's also a lot of sediment that's coming down from the Great Barrier Reef itself. So as a marine geologist, we're very interested in, I guess, teasing out the different kinds of sediments that both come from above, this pelagic sediments, versus the shallow water coral material, halimita. Okay, looks like we've got ourselves some rock.
Yes.
Hey, this is what we want to see.
Okay, now we're getting serious.
All right, now we're getting serious. This is Scott talking about. Oh, and we've got ourselves a nice sea pen. Is it a sea pen?
It's
Jeremy?
No, it looks like, let's zoom in, it looks like a black coral.
Black coral. Jeremy, take over, please.
Oh, mate, jump in. All right. Well, we'll just quickly take a look at it. And I mean, so can we look straight in front of it? Yeah. Come out around the sides, look head on. So it looks like it's in the family Schizopathidae. So the repeated branches, or what we call pinnules, seem to be sub-opposite, which means one coming just slightly above the other on opposite sides of the stem. So this could be a Bathypathes, although there's a weird branch in the middle that shouldn't be there. It might be a mutation. It's very odd to have one anterior branch among the anterolateral branches. This is the first one we're seeing on this expedition, so it's really exciting. I've been looking for this one for a long time. We found a lot of these. So years back, there was the Sedaris expedition that dredged corals in the Coral Sea. And we found, and those corals were deposited in the Museum of Tropical Queensland. And we found specimens that look something like this, although nothing nearly as spectacular and obviously as in perfect condition as we're seeing alive here. So this is an amazing opportunity to, so the pinnules are sub-opposite, and so it's probably a Bathypathes. Could be Petula, so there's a lot of work going on among the species in this genus. There seems to be a gentle change in the suite of morphological characteristics between one species and the next, and what that means is the taxonomic boundary between these species are just unknown, but we now have this new molecular technique. We're using conserved elements that takes these unchanged regions of the genome and compares them between species, and the regions on either side of these conserved regions are quite variable. So we use those to compare different species to understand the evolutionary history. So I'm just going to look up. It's a little light. Could be. So there's a really weird, that branch in the middle coming off of what looks like the stem is odd. So I'll take a close-up view.
It's kind of a hint of some strain.
You could see how big the polyps are. So these polyps are probably, like an inch in length. They're very big. These polyps are huge. Normally polyps are, they could be less than a millimeter. These are very big. Often the deep sea black corals tend to have larger polyps. And
Still a little too bright, huh?
it's okay. So yeah, this one branch in the middle is really, really odd. It's probably a mutation, but I mean, we're gonna have to collect it to see what it looks like. So we have the lasers on it, right?
Yes.
Okay. I mean, this is a beautiful colony. You could see that the stem, the unpinnulated section, the section of the stem that doesn't have pinnules, is very short, and that's also very characteristic of the species. Sometimes the unpinnulated section can be 3 times the size of the pinnulated section. So these are all different characteristics that we look at. Okay, enough staring at it. Let's grab it. Yeah. Oh, can we get the lasers on it for 1 second, just to?
Oh, I'm sorry. They're not. They should be on.
Are they down?
Zoom out a little bit. There you go. Keep coming. Keep coming.
I'll give it one bit of a tap.
There they are. You see them?
Yeah. Oh, so this is really big. So those are 10 centimeters apart, those 2 green spots. So the longest pinnule could be 60 centimeters, something like that. Okay, let's grab it. Oh, amazing. So
really good picture of it real quick.
I mean, yeah, do your thing. It's a really short. The stem is very short, and the pinnules are essentially almost the length of the stem.
Good job.
So usually you'd expect it to have a nice long stem to get off the substrate, but again, it's characteristic of different species. So if this pinnule is actually consistent with the species, then it's absolutely an undescribed species. If it's a breakage and a regrowth, then it's just that. It's either an undescribed species with a mutation, or Abathes, maybe Petula, or something else that we haven't yet described. Yeah, this species is absolutely a target for this expedition. A lot of they do occur deep, so I guess this is the one dive that we would most likely expect to find this on. So we're really lucky that we found it. Okay, I'm going to give the reins back to... No? Okay, I'll stay for a minute.
Where would you like to grab it at?
In the back. Bio box 3-4.
The whole tank?
Yeah.
The whole thing?
Oh, yeah. Try to get at the base, whatever the base is, if you can. So I'll take this opportunity to explain why we're collecting the full colony. Well, besides the reason that this really could potentially be an undescribed species, and considering that we're really redescribing a lot of the species in this genus, this specimen will directly go towards what we call a holotype, or a paratype, some sort of specimen that directly is used to describe the species. Additionally, to identify the species, we need to look at the spines, the skeletal spines that occur on the skeleton of this colony, and those skeletal spines could vary from the top to the bottom of the colony. And there's also a feature called striatum, which are these small grooves at the very base of the colony. What's the skirt? The skirt. I think it's just the pinnule, the polyps you're looking at. So they're just fully extended and super long, especially near the base. You could see that they're it's hard to tell how long they are, but
So I have a question for you, Jeremy.
Yeah.
This is a hexacolor, right?
Mm-hmm.
When you describe the polyps,
yep.
how do you describe the 6 tentacles on something like that being so long? Where are the tentacles?
Yeah. So there's one tentacle at the top of the mouth. The polyp is the feeding mouth. One at the bottom, and then 2 on each side. And the 2 on the sides are longer. Huh?
One for you?
Yeah. Try to pull it up to get the whole thing, if possible. Yeah. So the 4 tentacles on the sides are, I think, primarily used to feed, and then the other 2 have a different function, and they're different lengths.
So that there is one polyp.
Exactly. Yeah. So each grouping. Amazing. So you could see we got the basal plate, which is the attachment point of the coral to the. So now we have the chance to look at that one branch. It will look up a little bit, just to see the starting point. Yeah, stop there. So that one weird branch is coming off of another pinnule. So it's probably safe to assume it's just a breakage and regrowth. Yeah, beautiful, really gorgeous. Dennis Apresco will be excited to help identify what this is.
Wow. It's really...
really. Well, this beats dredging. This beats bending sleds.
Oh,
And so just to reiterate the importance of collecting this full colony, it's such an odd-looking specimen that it very well could directly be used to describe the species. But if we don't collect it, there's no way to describe the species. And of course, this fundamental information about what species live in these different habitats underpins all the conservation decisions that we make. So taking this species allows us to make much more informed conservation decisions that thereby protect overall biodiversity in these, never before explored habitats.
Yeah, go ahead.
It's beautiful.
Yeah. Yeah, this is the start point. Ben, did you move that start point for the... No. So yeah, this is the actual start point breaker. This one we put in this morning. It looks like that accidentally got touched in. Here we go. Okay. Ready, just.
Yeah.
That up.
If you could close this other gate first.
Gate.
So the
Gates.
the other
Goes down a little. Can you go down a little more?
Are we taking a rock sample of the base here?
Yeah, we're gonna try.
Well, we're going to tap it.
Okay.
Right there. Right there. Yeah.
Okay.
Okay, let go. So it should drop. It's flight wall.
All right. Amazing. Ah, so exciting. Nice. Perfect. Man, that's it. So you want to go up? Look up?
Yeah, go ahead.
Yeah, we're done. We're going to tap the rock.
Just give the rock a tap and see what.
I'll give you a zoom in.
See how hard it is.
Tippity tap tap down.
Yeah.
Tappity tap, tap, tap.
Straight into tap.
We're certainly in our happy place here. That's hard. Yeah. It's pushing the ROV up.
It's really interesting.
Yeah. It's not a great color.
See if they're in there.
Yeah.
Oh, it's
Big mate today.
flamingo tongue.
Should see more of this.
What's the flamingo tongue?
Oh, it's a barnacle.
Barnacle.
Yeah, it's a gooseneck barnacle.
Gooseneck barnacle.
And it's a big one too.
Yeah.
Can we get it?
Well, let's get the rock. Let's do the rock
See
thing
if
for
we
now.
can get the rock
That's a
barnacle on
gooseneck
it.
barnacle. And
Yeah.
when they break up, they leave this really classic, strange, almost dragon claw style plates, right? So a barnacle is a crustacean. It's made up of various plates, and this is the soft part of it.
Oh,
Yeah.
The base. This is soft. It's rubbery, like or leather. And in fact, this is live. You can see the.
Yeah.
This is the mouth parts of
I can see
the
it.
crustacean coming out. Oh, and there's a squat lobster just under him.
Scale worm right up top.
A scale worm.
Can you put? I've got the lasers on. I just get a sense of the size of this.
Yeah.
It's probably quite big.
You want me to? I can pick up and move over there.
So it's 10. It's maybe 8 centimeters.
Yeah,
Seven?
decent size. So it's a crustacean. So it has a carapace, and the carapace is made up of plates, and they're joined together, and so they open up like a purse, and the mouth parts, the filter feeding. And that's what we can see at the moment. So the main part of the body, of course, is sitting inside those plates. But when it dies, these plates all disarticulate, they break up, and we've found them in the sediment here from rock dredges. I've never seen a live one at this depth. Reminding everyone, this is the deepest the camera's been in this part of the Great Barrier Reef, and so this is really fantastic to see it.
When we collected on the Investigator last year, it ended up being a new bathymetric record for the species.
Yeah.
That's right. And now another bit of trivia from the plates that we found, these barnacle plates, which were obviously dead, they were just fossilised. They were a species that were more common in New Zealand. So this goes back to what Dougal was talking about earlier, where the water mass here is very cold. It's Antarctic intermediate water. It flows from south of Australia, moving up the east coast of Australia past New Zealand. And so I guess it's not surprising we're seeing animals that have more in common with temperate New Zealand than we have here with the tropics, because the water mass has flown, it's basically come a long way from much further south. This goes to the heart of what you're doing, isn't it, Jeremy, with trying to understand biogeography, how species link across these big ocean basins, across thousands of kilometres.
Yeah.
You can see the animals, it's pulled its the filter feeding mouth parts back inside,
but this is a living gooseneck barnacle. Now, if you've ever been beachcombing on, certainly in tropical areas, you can find on floating logs and things like this. These are gooseneck barnacles. There's different species to these ones, but they can actually, they attach to drifting objects, and and then they travel all around the world's oceans on currents. But this is a benthic one. It needs to attach to the seafloor, and I think it's really quite amazing because they're very big.
So we're going to have a go at the rock and barnacle and bun.
Yeah.
I'm excited to see what new information this specimen provides. This, offers.
The name comes to me: Silapus fosteroi, identified by the crustacean expert at Western Australian Museum. What's her name?
Hey, Rob.
What's her name?
Oh, right. What do I get if I pull this off?
You get one red horse.
Yeah.
A Filipino beer.
Red horse.
And it was the first record for Australia.
Yeah, flamingo tongue is the common name.
Flamingo tongue, yeah. Gooseneck barnacle is another name. Gooseneck comes from some old tale that geese were derived from the sort of rubbery necks of these things, and somehow it sort of was collectively, these animals being collectively known as gooseneck barnacles. Of course, they've got nothing to do with geese, but fascinating animal of the deep sea, and for me, really interesting because it's the first live one I've seen. We found lots of the plates, they break up. We found them in the sediments in the Queensland Trough at around 1,200 metres, but it took a crustacean expert at the West Australian Museum to really to put a name to a face here.
Yeah, and it's in our current paper.
Here's a question: why did that big coral look yellow inside the box when they red first colour? So it's about talk about coloration.
Yeah, well, so the black corals could be any number of colors. It could be yellow, white, red, pink, and I guess the color looked different when the light was shining on it, and now when it was in the bio box and the sunlight was kind of at an angle away from it, it looked a different color. It is often hard to tell what the true color is, although the first look we got at it probably was the true color, but we were kind of approaching it. But when we preserve it, when we bring it, we'll preserve it and we'll describe the color of the tissue because it does change as it dies. So it is an important thing to distinguish because it could be a species-specific characteristic.
Yeah, the other thing is the ROV itself has numerous lights all over it, and we've got this quite bright light in front of us, and you might notice occasionally they do a white balance check. They just check that the white balance on it is correct, so you're getting true white and not some dull pink or something like that. So you have to calibrate your lights quite regularly. The other lights that are pointing into the bio boxes may not be white balances, so you're getting this kind of differentiation in colour just depending on what light is being shown at it.
Cool. It's amazing how much you see when you zoom in on a section of otherwise bare rock, right?
Yeah.
Life will find a place to attach.
But you'll notice the dark rock itself. I mean, the dark is actually ferromanganese oxide. It's natural in the deep ocean, and certainly on the last voyage, as we did these long vertical transects, you notice the deeper you were, the darker the rock. There's more staining in the deeper waters, and as we went up the sides of those coral reefs, that staining would slowly disappear until you got the true colour of the exposed rock. But down here at around 1,800 metres, it is this dark stain, and out in very deeper parts of the ocean basins, the abyssal plain, 4,000 metres or more, you get this crust forming, ferromanganese crust that are these nodules. They build up on the seafloor. In fact, there's quite a lot of interest in wanting to mine them because they concentrate precious metals within the ferromanganese crusts. So we're going to have another go at getting this gooseneck barnacle. It's probably around 2-3 centimetres, about 5 centimetres long. But yeah, it would have quite a decent attachment to the rock. But the rock itself looks like it's very hard. We've already had one go at it, and it didn't budge. Of course, as marine geologists, we're really interested in the rock because it tells us a lot about the origin of these canyons, and we're sort of coming up with ways of how we might collect these rocks, and including, even possibly doing a drill. There we go, and a good pluck. Thank you very much, Ben. And looks like it's pulled off a bit of rock itself, or it might have just exposed it. But you can see what Ben's actually clasped onto is the stalk of it, and this is leathery, like you could actually bend it if you held it in your hands. There we go, you can see it sort of squishing. It has these sort of classic sort of ribbed shapes on the side.
Perfect figure for a paper.
Yeah, now if you look at the top, you'll actually see the plates themselves, and there's sort of some growth around the outside. But there'd be at least 4, possibly 5 plates making up each half. And when they...
Where would you like this one?
1A, please.
1A.
And when they break up, they have really interesting shape. So the first time I saw these in a rock dredge, they were stained dark from this ferromanganese oxide, but they almost looked like dragon's claws, like big sharp claw shapes. But all we were seeing was just individual plates from this gooseneck barnacle that had broken up. Thanks very much. Got Ben and Cody on the ROV controls. Thanks, gents. And John backing them up, running with the YouTube feed. Got lots of people here in the control room. Jeremy, Scott, Alex, Marty, Stephanie. And of course everybody watching from on shore. And we're really pleased that you can join us here on the ROV Falkor. We're on the Seamounts, Canyons, Reefs of the Coral Sea voyage. It's like almost an extension of the visioning of the Coral Sea voyage. But here we are on the edge of the Great Barrier Reef in a canyon draining the Ribbon Reefs. So the Ribbon Reefs are a line of barrier reefs which run northwards of Cairns for a decent distance, about 80 to 100 kilometres long, and we're opposite one called Ribbon Reef 5, and so there's this really large canyon, and we're just starting to come up the sides of it now. So think of it like, imagine if you're standing at the foot of a very large U-shaped valley, and you're looking up the sides, and so that's where we are now. We're just starting to work our way up the valley sides. The gradient increases. It's not super steep. I think the highest gradient we've measured here is around about 25 degrees. But if you're in a car, you could probably drive down this slope. And we've left the valley floor behind, and we've just got this sort of gentle upward gradient.
That's pretty vertical almost. It's gradient.
Special shout out to Arlo.
Yeah, okay.
Thanks for watching.
Straight up.
And we've still got plenty of marine snow out there. This has been a real feature of this dive. We just saw it literally all the way down through Dougal's dive earlier in the day. And of course the seafloor here is really muddy. Not devoid of animals, of course. They're definitely there, and we've been seeing echinoderms like sea stars and brittle stars. We've seen quite a few sea cucumbers, no surprise there. The vacuum cleaners of the deep. You know, their food source is this mud, and they leave these really distinct fecal trails behind. Keep an eye out these burrows too. We're seeing the occasional burrow. Now, I mentioned earlier they got this. We have seen giant isopods earlier in other parts, earlier in the other expedition, and of course the evidence would be seeing a live isopod inside. So just keep your eye out for them. But look beyond just the moonscape, and you start seeing lots of detail there. Like, well, certainly like these beautiful swimming scarlet-coloured shrimp drifting past the screen. But yeah, it's the background. Get your eye in, you can see there's mounds, trails, of course, probably left behind by sea cucumbers, like dots of burrows, and you know what is that? Who knows? But there's a whole science behind what makes these trails, and you know, we don't have answers for all of them. Some are quite obvious, you know, like the marks left behind by like sea stars and brittle stars. You know, have this kind of classic 5 pentaradial symmetry patterns on the seafloor, but other things are just complete mysteries.
Alright, so we're at the beginning of the transect part. I'm about to set up and get ready. Thank you.
Thanks, Cody. Alright, so we're just about to do a 500 metre transect. This has been an important part of this expedition, where we were getting standardised length transects. We choose them ahead of time. We don't intend to stop during it, so this is a drifting dive over 500 metres. If we see anything really spectacular for Jeremy, we may come back, but the idea is not to stop. And what we have, we've got Alex over in the analysis chair. She's using a program called Squiddle, and we've got Marty in the event logger, and the idea is that we really systematically identify everything that we see, and even, you know, that we don't see. Of course, absence is important as presence, and so these 500 metre transects we're doing every single ROV dive, and of course we're doing lots of sampling as well for the biologists. That's really important. That
How do you like the lighting in the camera angle?
Okay.
What's that?
Reduce the shadow a bit.
Okay.
That's good now.
Is that better?
Yeah, thank you.
Perfect. Pretty eventful before we even get to the real
Yes.
incline.
Okay. So are we underway?
In about 5, 4, about right now.
Thanks, Cody. And we're off. Okay, so this is the 500 metre transect, and we've got people logging away over in the corner of the control room. Oh, this is interesting, Jeremy. This is another one of these siliceous sponges, exactinella sponges. This is a stalk, and we've probably got zoanthids growing up the side. And we did collect one earlier. This is a sample we kept for James Reimer. Hopefully he's still watching. And James is a zoanthid expert, so he might be...
It's a speed, all right, guys?
Oh, yeah. Big sea star over there on the right-hand side of the screen. Yeah, decent size too.
And we're just going straight to that finished waypoint. We're not going to follow like waypoint 3 to waypoint 4.
Yeah, no, just straight through. It's fine.
Okay.
Okay, so this is the sides of the valley that form the edges of this canyon. It's not super steep, but it's just a gentle gradient uphill. You know, it might be around probably anywhere between 10 and 15 degrees kind of, real angle. Still quite muddy. We saw a rock exposed outcrop at the very base of it, which is really cool for us. We're quite curious as what is making up the size of these valleys. Is it just a great big mound of sediment, or are we seeing rock with just a veneer of sediment over the top? So it's not until you actually put a camera down can you really make those kind of judgments. We've mapped this area really, really well over the years, so we have a very good understanding of the shape of the seafloor, but not necessarily what it's made of.
Seagrass?
Yeah, I'm not sure. Maybe something dark and long.
It could be just part of silicious, like a sponge spicule.
It was very dark.
Yeah.
Something like that. I'm going too fast, just let me know. If you need me to change something, let me know. I can change for you.
No, more carbonaceous. In this part of the world, it's tropical, and so most of the plankton that's drifting down from above tends to be calcareous. So calcareous plankton, foraminifera, and if you combine that with the carbonate material the limestone material that's coming from the shallower Great Barrier Reef, which is only 8 kilometres from where we are, most of this is calcareous. That's not to say there isn't silica in it. There's, radiolaria. Dougal Lindsay talked about seeing radiolarian in a water column. So there's definitely siliceous material here. And of course, we've got benthic marine life like siliceous sponges that are pulling the natural silica out of the water column itself to make up its long spicules. But, probably between 80 to 90% of this mud is made of calcium carbonate or limestone. Contrast that with Antarctica, where it's really, really dominated by silica, through diatoms and the like. And if we were doing the same thing down in Antarctica and looking at the mud there, it would definitely be siliceous ooze. Not a lot of carbonate. It'd mostly be silica. But in the tropics here, it's definitely carbonate. Yeah, so I am part of the Trace Fossil fan club, but if we actually go back through geological time and look at rocks, like hard lithified rocks, old geological rocks, there's plenty of evidence of marine life, like mudstones, for instance. You can see these trace fossils left behind, as fossils within the rock. The animals have long since disappeared, but you can actually see the fossils left behind. So lots of people are very, very interested in both, the living trace fossils that we see on the seafloor, what is there presently, versus what you see in that deep geological time locked up in hard rocks. So it tells us a lot about past marine life, what the environment was like. So if you see trace fossils left in a rock, you can tell straight away it's probably a muddy environment, and lots of epifauna, marine life, that sediment feeders. These are, or deposit feeders, animals that are, that feed on this kind of this ooze and mud for their nutrients. There is a really good fecal trail that's probably from a sea cucumber. You can see almost like a tube of toothpaste left behind, but because this seems to be so undisturbed, this material just seems to last for a long time, until some kind of, maybe a storm event or even an underwater landslide disturbs it. It'll remain there until something bioturbates it and turns it over. So we can see lots of old mounds, at various different stages of decay, where they're being turned back over into into the seafloor itself. So, it's a really fascinating environment to look at, because we're looking at the past history of animals, even though they're long since past. Now, I would say the parts of the seafloor that are more white in colour, they're probably the more recent ones. So that's probably a decent hypothesis. So white is fresh. Mottled grey tends to be more old. So we're now at 1,700 metres, so 1.7 kilometres down, and just to tell people again, we're on the ROV Falkor on the Seamounts, Canyons and Reefs of the Coral Sea expedition, and this first part of leg 2 of the expedition is going to be focused on the Great Barrier Reef margin, looking at the deep Great Barrier Reef. In about a week's time, we will mobilise over back onto the Queensland plateau where we've been previously, and have a bit more of a deeper look at these really fascinating ancient coral reefs out in the offshore Coral Sea area. So we'll head out to Osprey Reef as one of our target sites. But for now, we're on the deep Great Barrier Reef, and it's really quite unique. This is the deepest any camera has ever been in this part of the Great Barrier Reef, and certainly no camera has ever been down in the canyons themselves. And for me, it's been a long time wish to be able to see what's living down in these amazing canyons. They're so rugged. They're up to 20 kilometres long. They extend out from the edge of the Great Barrier Reef down over 2 kilometres deep in places, and they're just an amazing relief. They're incised up to about 600 metres in places. So imagine if the continental slope was just a clean, flat surface. These canyons have actually cut down, incised into the seafloor, up to 600 metres in places. And they start right at the very edge of the Great Barrier Reef. So the heads of these canyons are in about 100 to 200 metres, right up hard against the edge of the coral reefs. In fact, we call these shelf-incised canyons, because literally they're very mature. They're quite old, we believe, and the heads of the canyons start almost at the edge of the reefs. You could stand on the edge of the Ribbon Reef and look down vertically in the tops of the heads of these canyons. And then they run almost 20 kilometres further out into the Coral Sea, down to about 2 kilometres depth. And we're now just going up the
Can we pause transect real quick?
Northern side.
Pause transect?
The southern sidewall of one of these canyons as you're looking at westerly towards the Great Barrier Reef.
We just got to do some vehicle management real quick.
Yep. There's some gambling occurring on that critter from the beginning. We'll get back to
Serianthid. Okay.
Let's see.
Just repositioning the ship, are you?
No, we've because of the currents above us, it's creating this huge sail in our umbilical, so it's doing initial.
Right.
And when we get a sail, we tend to porpoise.
If we need to settle down, there's a coral right in front of us.
I'm heading there. If it was any faster,
looks like no, maybe we're going in.
Just doing a bit of ROV management here. We've got a fairly decent current running northwards over the canyon, and we've got a fair bit of the umbilical cable out at the moment. So the guys are just adjusting the amount of line out so we can better control the SuBastian. Of course, every time we stop, we kind of stir up the mud. We've seen this already. It's fairly soft seafloor, so we have to kind of manage how we move across the landscape so that we're not we're trying to avoid having all this mud wash up in front of us. But if you be patient, we'll be clear of it very shortly. We've got Julia Summerling online. Hello, Julia. So Julia is one of our fan favourites. One of the world's most expert divers out from the Coral Sea. She's probably spent more time underwater out there than anyone on Earth. But she works on Mike Ball dive expeditions, and they do a lot of diving, expedition diving out in this part of the Ribbon Reefs. And she's asked me if we're going up to the Starkey River channel, and the answer is yes. I think more probably around mid of next week, we're going to head further northwards to a big canyon that sits between Ribbon Reef 9 and 10. It's the very northern part of the Ribbon Reefs. It's one of these shelf-connected canyons, so the head of the canyon actually cuts right into the shelf and is connected to the shallow shelf itself. And the thinking is that it's the older Starkey River. And Starkey River is an existing river on the Queensland coastline, and of course it finishes at the coastline, which is about 50-60 kilometres west of where the head of that canyon is. But in the times when sea levels were lower, down around 100 metres lower than what they presently are, that Starkey River would have actually debauched, or actually, the end of it, the mouth of that river, would have ended right where the head of this Ribbon Reef 9-10 canyon presently starts. And so if we have enough time, we're going to do a bit of some ROV dives at the head of this canyon. So we may not get as shallow as you'd like there, Julia, but we'll give it our best shot anyway.
See how large it is.
So you can imagine, we're down nearly, not quite 2 kilometres, but about 1.7 kilometres down, and with a fairly strong northerly current. It's a bit of a delicate dance. The ship at the surface, the ROV, 1.7 kilometres down, and then you've got all the strong current down through the water column, and of course the ship has to keep pace with where the ROV is going. We're moving in a southward direction up the southern sidewall of this valley, and so we're basically moving against a northerly going current. So it's always a bit of an interesting dance between the ROV and the ship above. But the guys here do a fantastic job. So really privileged to be here. So just to remind everyone, we're on the northern Great Barrier Reef margin. We're going up the southern sidewall of this big U-shaped valley. We did a fairly long Dougal dive down through the water column.
Begin transfer.
And then we traversed across the very, very flat channel floor of the canyon, and now we're moving up the sidewall. Good question about these from worms. Good question. So if I saw these mounds up on the shallow lagoon of the Great Barrier Reef, these would be likely Callianacid shrimp mounds, right? These are burrowing shrimp. They live in the lagoon seafloor, but down here, don't really know. So remember that some of them are quite old, right? So we're seeing the accumulated remains of them. I would say white is recent. The dark grey mottle is probably older, but until you see an animal popping out of it, you can't really be sure what's there. Certainly this is a zone where you do get worms, mud worms, like Echiurine worms is one type of worm. They've got this really classic T-shaped proboscis, very long proboscis, out on the coral sea reefs in this deeper part of the flanks of those reefs. We did see quite a few of these Echiurine worms, really, obvious, have almost a Y or T-shaped proboscis. Other worms that you find in this environment are acorn worms. They tend to be more, your classic worm shape, but they leave these really obvious spiral type fecal pellets, the poo, worm poo, is spiral. Now, I haven't seen too many of those yet. So, it's, I said earlier, there's this whole science around trying to understand the marine life that creates these trace fossils, these Lebenspuren. And some of them are really obvious, and others, we're still guessing. The people who spend their whole lives chasing this elusive animal that's left some strange mark on the seafloor. I know for me that the really weird one that we've seen out in the Coral Sea have these sort of parallel imprints, and they're burrows, they're holes, and they're parallel holes about 1/2 a metre apart, and they're in clusters of them. So you'll get 6 or 8 of these parallel holes in like a print on the seafloor, and then they'll occur again and again. Now, we have no idea what's making those.
Just updated your position, Jason.
So everyone, really, it's really exciting to see the sorts of, here we go, I was just thinking on the right here, one of those sort of parallel burrows. But so far we've seen these sort of strange round imprints on the seafloor, one at the very bottom of the screen now. You can see a fecal pellet just on the left-hand side, probably very likely a holothurian, long since departed and left its poop behind. The vacuum cleaners of the deep. Of course, Jeremy is really interested in his corals, so typically we find our corals attached to rock. It's not quite the environment we're going to find corals in, but we've picked up an absolutely beautiful black coral before. Anyone who was watching about half an hour ago, we picked up beautiful Antipitherian entire colony, and it's now in the bio box, and we'll bring that up at the end of the dive. So still lots of, mobile marine life. We've got shrimp, these lovely scarlet shrimp. We've got some big purple sea cucumbers, these probably Benthodites. We've got these smaller, delicate pink sea cucumbers. Before this voyage started, I reached out to a lady who's a sea cucumber expert at the Museum of Victoria, and she was involved with the previous RV investigator voyage into an abyssal plain.
We're going to have to pause transect again?
Yeah.
Abyssal plain study much further south from here, quite deeper depths, and she was able to identify 3 or perhaps even 4 different species of deep sea cucumber from a drop camera that we had on the RV Sonne, which is the new German research vessel. We brought out the Sonne to the Great Barrier Reef back in 2017, led by Maya Moktadi, a paleoecologist from Marum in Germany, the German Marine Science Institute, and we collaborated together. We were fortunate they had a deep sea, well, it was a drop camera, so it's nothing like an ROV. It was basically a downward looking drop camera, and we got down to around about 800 metres at the head of this same canyon that we're on now. That was the deepest the camera has ever got in this part of the Great Barrier Reef. We only had it down there for about 20 minutes, so it was only very, very tentative. But we drifted over about 3, maybe 4 different species of sea cucumber. Yep, and so from Jamie B on YouTube, I agree with you. I really enjoy looking at sea cucumbers too. So for us, it was a big surprise when we put that drop camera down from the RV Zona just to see how many there were. Of course, it's no surprise once you get a bit of handle on the environment. You know, it's clearly very, very muddy, and it's perfect food for a mud-slurping sea cucumber. But, you know, when I do a lot of scuba diving on the Great Barrier Reef, you know, in the shallow waters.
All right, Marty, we're going to go back on it again.
And, you know, sea cucumbers are a really common part of the Great Barrier Reef, the shallow Great Barrier Reef I'm talking about. And because they come in lots of different colours and shapes, and they're really quite an amazing animal. I quite like giving my kids a fright when we're out snorkeling. I'll pick one up and, you know, they squirt out their tentacles. These are called Cuvierian tentacles. If you get the tentacles on you, it's like chewing gum. It's really hard to get off. I don't recommend it. You know, you need to know you need to be aware of which end you're picking up. But yeah, they're very, very numerous, and in fact, you know, they're even harvested. They're commercially harvested. There's a well-regulated fishery on the Great Barrier Reef that harvests sea cucumbers. Slug divers, we call them. They collect them and they're used in the.
If I go slower, it's better.
Okay. They're eaten in restaurants in Hong Kong and China. So we're just passing a large pink sea cucumber on the right. And actually, we've got a fairly decent gradient here. Yeah, this is good to see. You know, we're expecting. You know, we are going up the sidewalls of the valley of this canyon, and it looks like we've got an eel down here on the right. Now, I'm no fish expert, but you know, we have identified one of the eels in this part of the world as a grey cutthroat eel, Cynobranchus, I think, and you know they so we have got some IDs on some of the eels down around this depth, but we're certainly reaching out to any of the fish army experts out there on shore. You know, quite happy to be corrected here. You know, we're lucky to have Jeremy Horowitz on board, who's a black coral expert. Myself, I'm a marine geologist from James Cook University here in Cairns, actually, so I'm not too far from home. About 80 kilometres or so away. So this is lovely. This is almost like my backyard, but of course, you know, it's a strange backyard. We're now at 1.7 kilometres down. But from where I am, you know, I look out over the Great Barrier Reef, you know, the shallow Great Barrier Reef, but I've always been intrigued by what's beyond that, what's in the deeper parts. And this is the first real deep camera seen in this part of the Great Barrier Reef. So it's a long time dream to actually see what's down here. And if we think of the Great Barrier Reef, it's far more than just the collection of coral reefs that we can see from outer space. It's this entire ecosystem that stretches from the shallow mangrove habitats that rim the shoreline across the interreef area, the lagoon, which is around about 30 to 40 metres deep. The coral reefs themselves, and of course we've got coral reefs that stretch from this is interesting, trace forelines with zigzag. It's like they got lost, and there's quite a few here actually. Yeah, but you know, it's a real mystery how old are these. You know, unless you see the animal right at the head of it, you don't know how old it is. Are we still on the transect?
Yeah.
Okay, all right. So we are on a transect. It looks like there's an animal sitting right in the head of it. With some enormous arms. I actually, you know, my guess is it's probably maybe a pencil urchin, and we were just looking at the spines sticking out of the seafloor. Now, you know, urchins themselves, you think of sand dollars on a beach. Now, sand dollar is just a flattened version of an urchin. Oh, there we go. I think we got something interesting. It looks possibly a brittle star or a sea star sitting inside a giant isopod hole. Right, that's my call. Someone can try and correct me, but
Rabbit in a wombat hole.
yeah, rabbit in a wombat hole, says Scott. Yeah, well, we're in Australia. We're going to have wombat somewhere. But I'm going for an isopod burrow. But it looks like the animals, animals are making use of these new habitats. Like perhaps the isopod's long since gone, and now it's just a home for some other animal. But so we're on this transect, this 500-metre transect, and I was just talking about the collection of different ecosystems. So as you move across the shelf, you're going. In and around this matrix of coral reefs. We have our inner shelf reefs, we have mid-shelf reefs, and the outer shelf reefs is this barrier, a chain of barrier reefs. It's called the Ribbon Reefs, where we are now. And then we get to the edge of the Great Barrier Reef shelf, and then it's just a massive long line of canyons, and they stretch from around about Townsville farther north, and we've counted well over 100 individual canyons. We're right in a very, very dense part of these canyons, the Ribbon Reef canyons. Individually, they stretch up to about 20 kilometres out from the edge of the Great Barrier Reef down into what's called the Queensland Trough. It's sort of a narrow structural basin that... Jeremy, what do we got here? Is this a black coral?
No. Feather star?
Or a brasingid, could be a brasingid sea star. They have that sort of classic pink colour. Anybody who's watching from ashore who knows we'd really welcome then. So Dinah's asked, are there any pictures or diagrams out there of the different trace fossils? Look, one of the things we're going to do, and part of this voyage, is we are taking multiple photos. We're doing it through two ways of doing this. We have the Squiddle program, which is taking 5-second photos, so it's time-stamped every 5 seconds. We also have an event logger, so anything really interesting that comes up. We have Marty sitting in the hot seat of the event logger, and basically anything that's really interesting, we will take a snapshot and we'll put an ID, and it's captured for all time, so we can go back and revisit it. And one of the things we want to do, particularly for a colleague, Rachel Przyslowski from Geoscience Australia, who's also part of the Lebensborn fan club, that we want to put together a bit of an inventory of Lebensborn, or life traces, that we're finding here, and so we can start building up this kind of understanding of what the variation is. So we can have there's mounds and burrows and tracks and trails. There's sort of those kind of descriptive terms for Lebensborn, but ultimately we want to try and understand what animals are making them.
They make for a good coffee table book.
Yeah, that's right.
Yeah, classic coffee table book that you'd give your favourite auntie for Christmas or something like that. A bit of light reading. But of course, this has been done. When people were first putting cameras down on the seafloor, and we're going back to the 70s and 80s when, we're talking about the Americans here, and I forget the classic names. If you're into deep.
Yeah, keep going.
The deep ocean explorers from the 70s, we're talking sort of Ballard days and things like that. People who were putting cameras and submersibles down, and they were just trying to understand better. I guess it was coming back to plate tectonics and how the ocean basins were created, how young or how old they were, what's living there now. There there was a whole fleet of vessels, particularly the American ships, the U-Knowles fleet, who were just towing cameras, downward-looking cameras, at depths of 4 to 5 kilometres down, and just taking myriad photos and bringing the first pictures back of the deep, deep ocean. And you know, there's this sort of classic volume you can pull out from libraries, and it just shows, you know, hundreds of these black and white photos from cameras that were sitting in these, you know, chambers. You know, you imagine the pressures down, the enormous sea pressure down at these, you know, 5 kilometres, 4 or 5 kilometres down. You know, amazing pictures. And even now, so we're still trying to understand better what's living down here. So it's taken this long, to 2020, to get a deep camera down in, you know, this part of the Great Barrier Reef. So it just shows how long and how hard it is to, you know, to get vision down in these depths.
Just trying to push through this one. Hey, Mike, are we at a plateau right now?
Yeah,
Kind of. Feel it.
it's going to go up again.
And it's going to come up again. I can feel the current pull.
And James Reimer from Ryukyus in Japan, still watching. Thanks, James. You know, these obscure anthozoans. You know, I always defer to Jeremy here, but you know, we really appreciate people watching from ashore to, you know, help ID some of these animals. They're quite strange. I remember one really weird one. It was a harp ctenophore, like a benthic ctenophore. So, you know, Dougal would be familiar with these. But the first time I saw it, I was like, "What the heck is this?" You know, used to ctenophores in the water column, but when you actually see them attached to something, they look completely different to anything that's floating in the water column. We saw our first live gooseneck barnacle attached to a rock. That was really amazing to see. We've found the dead fossilised remains of the plates before, but hadn't seen a living one down here in these depths, so that was great. Yep, Dougal's saying rabbit ears. That's what they look like, but yeah, it was. We had to really reach out to all our helpers ashore to try and identify these things. Yeah, Jamie's saying benthic ctenophores too. So look, after we leave the Great Barrier Reef margin and we get over on the flanks of the sides of like Osprey Reef and some of these other coral sea reefs, we will be doing deep ROV dives. So there's every chance we'll get to see some of these quite strange harp ctenophores, and you know, I'm really thrilled that we can get the ROV down deep as well. So the AUV is not being mobilised on this leg 2 of the expedition. We've stopped doing that, and it's only ROV work and multi-beam work with lots of sampling, we hope, for the remainder of this leg until we finish in Brisbane on the 26th of August. 30th. Sorry, we've had an extension. 30th of August. So, yeah, so 2 weeks. We got 2 weeks at sea now. We've got rat tail. We've got a rat tail just hovering in the water column, and we've seen this really interesting behaviour that, you know, where it's head down with its tail up. Now, if anyone's watching from the shore can explain why that's happening. Now, there we go. That's great. So my guess is it's looking for food on the seafloor, and you know, it's looking. If that's where its prey is, you know, it makes sense that it's head down. But you know, most of the time we're just guessing here. We're just trying to figure out as we go along. So, you know, we're very open to advice from everybody watching.
Take position.
Makes sense. Range. Just updating your position.
Yeah, could be a hunting position. Yeah, it makes sense to me.
And being down on the ground looking out.
Yeah.
Not to be off.
Maybe something wombats. Wombat holes.
Yeah, wombat holes, yeah.
Oh boy, there it goes.
Now, this is a decent sized shrimp. Yeah, about 10 centimetres, maybe longer, yeah, nearly 15 centimetres. Lovely. And if you look to the left, there's another shrimp. We've got a burrow, maybe one of these big giant isopod burrows. Because what we want to see is a living isopod, and we have seen that previously. So we're driving up the southern sidewall of this very large U-shaped valley, which is the mouth of the canyon. All right, so we're about 8 kilometres due east of the Ribbon Reef 5 reefs, shallow coral reefs, so you could actually scuba dive on them. And I'm sure there's people watching who've done this, and I certainly Julia Summerling has probably done scuba dive hundreds of times on Ribbon Reef 5. One of my favourite parts is to dive in the Great Barrier Reef. And if you're very, very lucky and the weather is good, the dive vessels will take you on the outside of these ribbon reefs, and you can do a drift dive. The currents are usually quite strong, so drift diving you have to be dropped in the water, and then you'll have a tender that will pick you up at the end of your dive. And I've been lucky enough to do a few of these drift dives, and you're just basically floating across in front of this huge wall, almost a vertical wall of coral. And you know, being on the seaward side, it's got this lovely clean water, and there's all kinds of corals and the like. And you know, sometimes these big swim-through holes, big caves that have been cut into the sides of these coral reefs. You know, it's like flying along, I guess, the Great Wall of China or the edge of a big vertical building. So it doesn't happen too often. You've got to have the conditions absolutely perfect to do that.
How many meters? This ballpark, not directly to the finish line.
Yeah, that's right. We have our laser beams. They are measured exactly 10 centimetres apart, so that gives a good scale of what's going on here. I mean,
We've got about 100 meters left, and I'm going to go past it a little bit just because of the porpoise. Let's get you some better stuff.
Most of these mounds are between 10 and perhaps 20 centimetres wide. At this stage, don't really know what kind of animal we've got there. It looks like some sort of shrimp crustacean swimming past us.
Bring it to 150.
Yeah, good question. Can the lasers damage fish eyes? Well, we haven't seen. You know, we've been fairly close to lots of fish with the these are green lasers. Green being the, you know, they will penetrate further through the water column than perhaps say red lasers, which are absorbed much quicker. We haven't seen disturbance so far. I mean, if they're disturbed, it's probably more from the bright white lights that we're using, the floodlights that we're using, just to give us this view that we have now. But you know, down at 1.7 kilometres down, it is completely black. You know, if the lights were turned off, it's pitch black. And so all the animals that live down here have had to adapt to that complete pitch blackness, and they do this through a number of ways. They make themselves completely black by taking on almost a scarlet red colour, which is completely absorbed by any light they may have ever got down here. So you know, they're invisible to the eye, or they have amazing eye adaptations themselves. So they're looking for light emitted by other animals, bioluminescence, for instance. And we've seen that with some of the fish here. You know, they have really large eyes in comparison to their body. So they're trying to harvest whatever light they can get, be it ambient light or, bioluminescent light, where animals are disturbed and they send out flashing signals and the like. So, it's really an amazing dark world down here. Want to take over for a bit? Jeremy's going to take over the audio seat for a bit. I'm going to have a cup of tea.
All right, so we're just finishing up our transect here. We'll be done fairly soon. And so we're currently at 1,575 meters. It's almost 3 degrees Celsius, so pretty cold down there. Cold and dark. Hello, Lisa. So it's been fairly consistent, with this kind of heavy mud, sand, with these mounds. Similar looking critters, but then when we find some sort of substrate, some sort of rock, all of a sudden it's the available space for a hard substrate for a coral to settle on, which is where we found the black coral. So it shows when many species need some sort of hard substrate to settle on and grow. So we haven't seen many corals during this transect. And it's because they don't have a place to settle down. If there was a nautilus shell on the grounds, like we saw the other day, a coral could potentially settle on that and grow.
So his model is working.
Yep.
Yeah. Oh, here. I have to check. Yeah, it's close. Type her out again.
Almost there, yeah?
Yeah. Hitting like a brick wall right now with that current. It's coming over this edge.
Yeah, you can see how hard it is for us to move forward. We're fighting against the current. But it looks like that fish is just having a real easy time settling down.
Something interesting, Jeremy.
Find something interesting?
Yeah.
Oh, I think I can.
So we can stop.
You want to look at this fish here?
When we get to your marker. When we get to the marker.
Yeah, okay.
Yeah, I might just do it as
Yeah. So we're going to have the ROV arrest once we finish this transect. And we can, once again, look at the substrate, look closely, look for forams, the sea butterflies, the tusk shells, all the things that make up the seafloor.
I'm hearing the ROV out of the controls. They're planning to do. Is that standard? Have we done that?
Oh, I think they just need a rest for the boat to catch up. Or
Yeah, cool.
Yeah, what I'm gonna do is I'm gonna come up in the sea column,
Yeah.
come back some, because right now we got this big, huge sail behind us. If we keep doing that, it's gonna just keep ripping us, keep ripping us back, and we're not gonna get good video, or we'll have a hard time getting samples because we're just gonna start those up,
Okay.
Shouldn't take that long.
We could take a push call.
Oh yeah, we'll take a push cord.
Good.
Yeah, definitely, we'll take a push cord at the end of it.
Thank you.
looks like a sea cucumber, maybe.
Yeah.
We've seen a few of these light-colored sea
Yeah, I
cucumbers.
don't know how much fish is really good.
It looks translucent.
I'll get the take on it.
Anemone.
Can you do a quick measure of how far we might be?
Oh, that looks like
From
that sea cucumber swimming on the left there.
Tomorrow and the day after, I measured it earlier. Our aspect is one different. So,
Quite different person branches.
yeah.
how you gotta go into the front and stuff.
It's funny, Dougal's comment is being blocked.
No.
They may think that the name is like a term, like an offensive term. Careful, Dougal.
John was just telling me they have been blocking some comments back home, or back in the head office at Carly filters or something. So yeah, some people get a bit upset when we take samples.
Well, I mean, we do try to explain to them the reason, the rationale behind it.
That's right. That's,
Yeah, we can ask the ROV pilots to add a tickle mechanism onto the ROV arm. Sure, absolutely. I mean, that's a standard scientific technique, right? To tickle, see if it giggles. Yeah.
They normally poop their over there.
Right.
That's what happens.
Giggle and poop.
Yeah, they do.
We got the paintbrush must have made it.
Yeah, with the
The
brush.
bioluminescent.
Got arms.
Yeah. Looks like a anemone in front of us, potentially. Big anemone. Is that anemone? Yes.
The one right there in front of us?
Oh, no. It's a feather. I think it's a feather star.
Now, those wouldn't be called a basket star, right?
No.
Feather star.
Yeah.
Because a basket star is climb up to the corals, right?
Right, and they come out at night.
Yeah. Really
Yeah.
freaky looking.
We've seen a lot of these. They're pretty big. I mean, one of those arms has got to be a half a meter long.
If you want to stop hanging on, I'll just finish it up real quick. Yeah. Only got a few meters to do. And then we can sit down. Might want to get him to come start coming faster towards us.
All right, we're gonna take a push cord. Is that right?
Almost.
Okay.
We're almost there.
We're dragging considerable amount of cord behind us. It's up in the air in front of us, and then behind us, it's causing a lot of drag.
Like a parallel with us.
So we're gonna remedy that.
Hey, bridge, have a new position.
Copy that.
So you catch up with us while we're sitting down on the board table. Right where the next star, feather star is, that's where we stand. We're up. So we went an extra 20 meters. Stop it.
Oh yeah, they're talking about Narita's.
Oh, K-Bus. The kitchen brush of science.
Yeah. We should have asked her what special tools to bring on board when we first arrived. I'm sure she would have mentioned it.
Oh, okay.
The K-Moss.
Just scrubbing down.
The tickle fingers.
We're good on the squid that we're getting. The squid, we'd go into the squids with the K-Boss and grapple in it. Then we'll get a piece of flesh from it on the actual brush.
Amazing.
They can date it, say what kind of species it was.
Wow.
And that is it. That's an extra 20 meters because of the issues. Sorry.
Thank you, Carney.
So you want to sit down or go up?
Yep, we're sitting down right now.
All right.
You want to push cord?
Yep, push roll.
Yeah, let's get it.
Okay, we have a core to push.
Let me just sit on down again.
Yeah. Are you being dragged back still?
It doesn't like this spot. Settle. Gotta dig myself in. Alright then. Watch up. If you want to follow him.
Yeah. I will follow.
Current is it coming?
Some people didn't believe me that I actually controlled some of the focusing on these last dives. No faith.
Can you pan right a little bit?
Yeah. Pan right, you said?
Left.
Left.
Oh, there we go. Look at that, those zip ties go.
It'll be number 4.
Coming out with the one. Next time, unfortunately. Number 4, Ben. Left forward is.
All right, the last push core we did was at 1.8 kilometers, and now we're at 1,560. Still under 3 degrees Celsius. And it's only 3:45. So we got time.
3:40.
We aren't ending early. We actually have a bit more to go. We're certainly not going to get to the surface. The aim of this dive is just to get to the top of the canyon, which is a little above where we are now.
1400.
At 1400, so we have 1,500 meters to go, which will take us to the next, 2 hours, 2.5 hours. We keep finding really interesting stuff, so yeah. It was funny, I thought for a second that we might not have enough, we might run out of time. Certainly not the case. I thought we'd get to the top and have hours to go. People laugh at me, deservingly so. Oh, look at that.
Nice.
Yeah.
Yeah, that's French kangaroos.
So if you'll remember, on leg 1, we never had one successful push core. Every time the sediment was, so coarse or dry, and it would funnel out once we picked the push core off the ground. But here it's so densely packed and it looks muddy. It makes for a really easy push core. Yeah, in these canyons we have sediment coming from the shallows, and it makes for a very thick and muddy sediment floor.
Okay. In the same area?
Yeah.
Awesome.
And you'll notice we have turtles on our bio boxes today, a pink one and a blue one, I think. Flipping away, flapping, helping us with the strong current. So the reason why the push cores are working better here is because it's a much wetter and thicker, denser substrate. So it's essentially like mud. If you were to stick your hand in it and squeeze it, it would feel wet and dense, as compared to leg 1 where it was extremely dry and there was just no hold to it.
Can you put me out?
You're not dating. I mean, there's no. I mean, essentially, you're going to understand.
Yeah, we could do. Potentially, we can date. Yeah, we'll just see what the condition of the core is when we bring it up. But it would be a short record in time.
Because it's constantly.
Geologically speaking, yeah. Maybe only 2,000 years, approximately.
For some reason, I just thought it was given away. Alright, good. Alright,
are we doing another one?
Yep. You can do 5 or 6, 30 species.
It's like a steam engine.
Yeah.
We'll do 6. 6 all right?
Yeah, it's good with me, which was easiest for you to access.
So, how do you decide where to take a sediment sample?
So that to scan this particular size. Okay. So in effect, what we've chosen this transect on this part of the canyon as a representative area of quite steep slope. We're after the steepest part of the canyon wall that we could see, and this was based on the existing multi-beam bathymetry data that we had before the survey. We mapped it again last night, in fact. We had time to do that just to verify and add a little bit more detail. So with a steep slope, we were hoping for some chance perhaps of seeing more rock outcrop. We have seen a little bit of that, but perhaps not as much as we might have liked. Because from a geological point of view, seeing the rock outcrop gives us a clearer understanding of the underlying geology and the history of that geology that these canyons are formed in. Also, over the next couple of days, we'll be in this canyon network, and we'll build up a picture of the whole canyon system by selecting this transect, and then tomorrow and the day after in some shorter transects, and all going well, we'll have a final transect or two on the head wall of the canyon right adjacent to the barrier reef. So that's going to be a really interesting location. You're welcome. No problem.
Yeah.
Right.
And see picking up the shadows behind the mountains.
Okay. Are these the biodilation mounts?
Yeah.
Really?
See the mountains there?
Okay.
So it's scanning in front for hazards.
Yeah, okay. Wow. For resolution.
Well, I just wonder if you could put that as an inset, or if that's
I'm sure they can.
just to show. I mean, this is really interesting. Like, we can see it visually, but even the forward-looking sonar is picking up those little shadows. That clearly, this area is very mounded.
Yeah. Very dense. We've done them in before. It's,
I wonder what the distance is outward.
yeah, I
The
don't know
sonar.
what those intervals are. 30 meters.
20 meters.
20 meters.
So
Okay.
it'd be nice to pair up a big mound with a big
Yeah.
shadow, I guess.
Yeah.
Yeah, because a bunch of those won't get those shadows, whether or not they have those mounds or not. The artifacts sometimes.
Yeah, it's interesting.
Yeah, because if you change the color, it's a little bit different.
Really. But it's still representative of.
In a way, because some of it's artifacts, so it's just a bunch of it's so high up, it's not pointing down.
Right up.
So that's pretty much backscatter stuff jumping off of
Okay.
what it can see, per se. But some of it is actual mounds. It's just it's hard to tell which one is and which one's not.
Maybe closer. Might be more.
Yeah, probably closer, like this one right here. So you see the mouse up there at the top right. Follow that one straight down. Right there. See
This?
that top one?
Or this?
That one right there. That's an actual mound. Now all that stuff to the left, that's in the very back end, that's all artifacts. Because it's not picking up anything, it's picking up blackness, void, and it's bouncing back.
Okay.
Now, if we had the blue view one, the imaging sonar, you'll be able to see each mound perfectly. It's just some of it comes in as artifacts with that
It's almost
scanning
laser
sonar.
beam density. Isn't it?
Exactly, yeah. Now, if I put it in ultra, because right now it's a medium, that's why we're getting all those artifacts. If we go into ultra, it's a lot slower, and all that will disappear.
Yeah, right.
So we use that as obstacle avoidance, per se.
Yeah.
You'd like to see it fast.
Exactly. Yeah, we want to see what's coming, where it's going.
Okay.
Yeah. But yeah, once you go in an ultra,
We're going to go in a cave.
Do you
Exactly.
have an oil petroleum background?
Yeah.
Okay, so in really turbulent zones, you have this blue view going where you're looking for pipes. Is that how you use it?
So basically, yeah. The blue view itself, this is the Sea King. So the Sea King's just scanning sonar. So you can see a shadow behind it. So if there's a pipe right here, you can see that shadow fall back behind it. You can see it's an actual heavy structure. But the blue view, you'll see the full 3D render of the platform or pipe.
So most of this artifact, is it?
Yeah.
Okay.
Now some of it's not. So like that one that you pointed at earlier, that one right there, that's probably not an artifact. And then some of the brighter ones, they're probably not artifacts.
Thanks, Ben.
Would that be? Could you guess? It's that one.
Well, this is 20 meters, you said?
Yeah, so that one's about 10 meters out.
So yeah, about 10 meters.
Yeah.
Yeah.
That's what the range on the side.
Yeah.
Maybe as we
put
move.
it in ultra, they're just asking about.
Well, it looked like it was picking up all of these mounds that we're seeing in front of us.
So that one.
Andy Carroll, good afternoon, mate. How are you? Andy was on leg one.
Andy, please
Go-
stop
go
clogging up
into
the chat.
ultra instead of me. Showing that the difference between some of the artifacts that the sonar produces. Mounds and all. You good, Ben?
Yeah.
And that's 20.
Bring it down.
Okay.
That's 10 meters right there.
So I'm just going to keep my finger on. This was it, right?
Yeah.
Hope that was real.
I'll be further out.
Okay.
Now it's 10 meters. I can go back.
Oh, it's 10 meters, so it should be towards the end up here. I mean, it's not a particularly large mound, so.
There you go. So
And
that's
you'll
ultra.
see the difference between.
So this might be
Yep.
a large mound.
And that's the shadowing gets cast black.
Yeah. Okay.
Cool. Just see how there's like a little pinnacle up there, and then you see the shadow on the back.
So that's your bridge right there.
Yeah.
Then that's the shadow.
Go back. So feather start. So we're off. So we're ahead.
Yeah. Right. Keep
At 1,500 meters.
Yep. Still under 3 degrees Celsius.
And still about halfway up the sidewall of this canyon.
Yeah.
Just go till time runs out.
This is reminiscent of leg 1.
Okay.
Yeah. All right, now we're gonna have to rush to get to the top. Oh, look at our.
Oh dear. Full marks.
That'll be there for years.
Well, between the next time we came up over this ridge and the currents just pushed right at us now. So we just finished transit, we're just heading straight up the mountain. I just flew back, we're painting and we'll try to get some of this.
You having a break? Yeah, go ahead.
We're going to sail. Yeah, we look up there, look at the top profile, 500. It's like 3 knots.
I'm on it. No.
Insane. Okay. So we got that big sail behind us.
So we're just doing a ROV crew changeover. Oh, what have we got drifting past us? There's a lovely Medusa coming into. No, it's a sea cucumber. Yeah. It's one of these beautiful benthodaites. They have this lovely swimming motion. So you can see the fringe at the front here. This is. I think the mouth just on one side, and of course the anus is on the other side. But when they're disturbed, they flip themselves up into the water column and almost do this beautiful Spanish dancer-like roof. That is just a fantastic view. Beautiful. You can see how soft it is, you know, like it is just this thin layer of flesh. Most of the inside is water, and then of course, whatever mud that they've slurped up. But we have seen them just like leap up into the water column, and that's pretty spectacular.
I have this turned up some, so that way we can actually move. It's pretty rough.
This shows how big the hole is.
Right, off we go. So was it?
Are we not going with boom or something?
Oh no, you go back on with them. We just finished the transect. I had them off for it to get those lightings back.
So we're in sampling mode again?
Yes, we're flying up and
there we go.
get away.
All right, so we finished our 500 meter transect and we're in exploratory mode. So whatever comes into view might be worth sampling. Certainly, definitely worth talking about. And just for scale, notice the 2 laser beams in the centre of the screen. They're 10 centimetres apart.
Hey, Jason, I got an update.
Still very cold down here. It's 2.9 degrees Celsius from the temperature mounted on the SuBastian ROV. At this depth, we're at 1,500 metres. We're well within the Antarctic intermediate water. So this water mass originated south of continent of Australia, and it follows these deep topographic basins. So it moves towards the equator, and right where we are, it's a very northerly flowing current, and we're actually flying into it. So we're going up the south wall of this large U-shaped valley. This U-shaped valley is the Ribbon Reef 5 canyon. So we're going up the southern wall of this, and it's just a gentle gradient up, maybe only about 10 degrees, real slope angle. So it's not steep by any means. If you're in a car, you could probably drive down it. But we started in the base, at the very bottom of the valley. It was absolutely flat, and then there was this quite distinct change in gradient slope, and we've been going up it for about 2 hours now, and our aim is to get to around about 1,400 metres depth, and it's basically the very, very top of this sidewall area. So no, water cannot freeze down here. Pressure has a lot to do with it. It's also tend to be more saline. So it's an interesting question. You know, if you were to take this water, of course, put it in a freezer, eventually it would freeze. But you know, even in Antarctica, you can super cool water. It can be like -2, -3 degrees Celsius. But it doesn't freeze, of course. You know, you're not getting great blocks of ice at the bottom of the ocean. It's just because pressure and how saline that water mass is, it always remains unfrozen. So what we have here is very, very cold water that's moving northward at the very bottom, the base of these canyons. And there's a depth that kicks in, and is around about 800 to 900 metres. So you may have heard Dougal Lindsay talking about it earlier. This is Antarctic intermediate water, and he's very interested in the sorts of plankton that live within it. Clearly there's plenty of marine snow down at this depth. You can see all these white particles in the water column. So we're flying, so just to repeat, we're flying up the southern wall of this canyon, but we're flying into a northerly going cold water current. What have we got here? Is this a black coral? Let's find out. Jeremy, what are we looking at here?
No. It's one of those feather stars again.
Is it? Okay. All right. I thought I might be looking at the different branches. So it looks like it's either a feather star or a Brisingid sea star.
What are people calling it?
Yeah, so the Brisingid sea stars have got this sort of classic sort of deep pink colour. We've seen plenty of them around, both on this dive and other dives. We just need a little closer look at them to really distinguish them between like a brittle star and a sea star, which is what a Brisingid sea star is. Starfish. Here we have an eel. My money's on a grey cutthroat eel, but certainly open to anyone from the fish army who can identify these. But interesting, they're pointing into, looks like they're pointing into the current, so maybe they're waiting for that, their food, their prey item, whatever that is, to drift along. So maybe that's an indication of the current direction that we're heading in. There's another one that's also pointing in the same direction. Oh, looks like a nice sea star over here. Oh yeah, really classic shape. Of course, they come in lots of different shapes and forms. We've seen them with very, very long, thin arms in earlier parts of the dive, but that's almost like your classic biscuit star. Yeah, where the arms are literally joined together by a big stretch of flesh in the middle. And we've got pencil urchin just over on the left-hand side. So, you know, this is the home of, I think, shrimp, echinoderms, so sea cucumbers, sea stars, brittle stars. These are all deposit feeders. Yeah. You know, they're basically, their food is in the sediment, or what is just drifting directly over the top of the sediment. And eels, we've seen quite a few eels here as well, and lots of shrimp. So, you know, it's definitely a habitat that's dominated by, you know, 2 or 3 groups of animals. We have been fortunate to see a really large black coral was attached to a rock. So, fingers crossed we'll see another rock. But my my guess is that we probably won't. We're going to see this sediment covered slope all the way up to the end, so we'll just keep our eye out for anything unusual. These great big purple sea cucumbers, Benthodites, I think is the genus. I think we saw one earlier. It was, you know, when you give them a bump, their defense is to just jump up into the water column and swim away. Alright, so we might have a feather star in front of us. Yeah, I think we're looking at a feather star. Classic 5 arms. So it would be a comatulid feather star. The comatulids have got these very small legs that they can attach to things. We had a really close-up view of one earlier. It was sitting on the sand. It had very short, sharp legs, a few centimetres long. It just attached to the sediment, and they can move around. They can actually walk, and they can also swim. They have some really fascinating cousins, the sea lilies or stalked crinoids. We have seen them attached to the sides of Osprey Reef and some of these other coral sea reefs. They're a real classic, ancient, almost relict fauna, because you can find fossils of stalked crinoids in limestone rock going back 400 million years. So, you know, I think they're just fascinating to see in the deep sea when you're seeing them live. Here's a fresh looking mound, so keep your eye out for that variation in texture, colour. Yeah, something's a bit more fresh there. Yes, James, we are on the lookout for stalked crinoids. This doesn't seem to be the environment for them. I mentioned that we have seen them on Osprey Reef and some of our other offshore coral sea ROV dives. So yeah, they're really amazing to see living. We're going to head out to Osprey Reef probably in about a week's time, and you know, we have very high chance of seeing them there if we're doing a deep ROV dive. That's right, James, you were at Osprey Reef with me, I think it was 2011. Well, we're heading back out there. You'll get to see the deep stuff. Yeah, I think we might have a bit of tilting in the vision simply because we are pushing against a fairly strong deep water current. Now, this is from this water mass, the Antarctic Intermediate Water, that's just flowing northward. So we're basically fighting against that. And of course, it's this delicate balance between ship and ROV. But when the ROV is down 1.5 kilometres deep, you know, it may not necessarily fly perfectly straight. So I did my PhD looking at underwater video, and I got quite used to having to, you know, tilt my head sideways and squint your eye and try and guess what it is you're looking at, because nothing was ever horizontal, nothing's ever perfect. You don't have this beautiful flat view across the seafloor. Oh, here's a great eel. Yeah, anyone from the fish army ashore who can ID this? And really big, fat, slow-moving eel. So I'm really curious, you know, what these guys are eating. We've seen so many of these in this dive. Oh, look at that, beautiful, lovely motion. Oh, this is a great view. You know, what is it they're eating? Is it the shrimp? We've seen plenty of that. They would be good eating. Are they going for something we have no idea about? It's got a good set of eyes on him anyway. You can see quite large eyes. It looks like he's reversing. Great swimming motion. Yeah, Nani says, off your day. Dougal. Sinapha brancus. Yeah, good. Yeah, that's pretty good. And we have one of these really big purple sea cucumbers. My guess is Benthodites genus, but pretty big, yeah, and really translucent. So you can actually see in through its body, which is quite amazing. So how do I do that? Right. Oh, yeah. So I'm just playing with the controls for the first time. There we go. Now, if you have a look, you can actually see its gut, in its internal gut, right? So the mouth is at the front, you know, where the larger frill is. The mouth is there. It looks like it's being disturbed. It's lifting up. You've got these papillae on the top, but then the gut is actually filled with the mud that it's slurping up, and then it's digested, and of course what comes out the bottom is sea cucumber poop, which is, and it's
zoom in on that.
Yep. All right, I'm gonna just have a bit of a play here. Oh, sorry, wrong way. Going down, and what do we got here? Yeah, it looks like a polychaete. Now, you know, polychaetas are Nemertean, and you know, they're multi segmented. Whoops, sorry, everybody. I have to get used to the controls. It's my first time.
Another one's back, Jeremy. Just a touch. Got another one up there?
Yeah. Oh, well spotted. Oh, look at that! What a view. You can see real detail in the skin now. You know, it's just like tiny little dots, little papillae, and then we've got these kind of larger, but we've got tentacles on top. You can see it was almost rhythmically pulsing. But yeah, this looks like a classic polychaete. You know, flat. You can see its feelers or proboscis in front. These are probably likely spines as well. But they very likely are animals on animals. Hey, Jeremy. Well, we've seen this so often.
Yeah. Can I get a manual zoom?
Yeah.
Go ahead.
Yeah, everybody's an ecosystem. I agree. But this is great. We haven't actually seen too many polychaetes so far. I'd say many of them are infaunal. They're probably in the mud. We're lucky to have this hitchhiker. We're getting a good look at this one. So I'll just zoom out a bit. Zoom out. And
click here in the manual focus.
It's on auto now. No, it's on manual.
Okay, so yeah, now try.
Okay.
Or do you?
Sure. Yeah, why not? We'll try one more. It's good to zoom in.
yeah, you're just getting a bit of swash from the ROV propellers here, just obscuring it. But anyway, you can just see this polychaete's just sitting on the, I guess, the fringe of this big sea cucumber. We're just playing with the manual focus here to see if we can really sharpen that picture.
think it's still on auto.
No, you're
Yeah. Okay.
cloudy. I can try one more time.
That's okay. And maybe you'll be better at it. You think you can do it? You can focus in on it? Sure. Give it a try. Yeah, it's tough. It's very fine-tuned.
Yeah, and if you have a look around the sea cucumber, these little white things, these are pteropods of various kinds. So there's a tusk shell, a scaphopod directly behind it. And I'd say these are completely everywhere. You know, we just need to zoom in to actually see them in focus. But, you know, it's this amazing animal. I'll just zoom out a little bit because I quite like looking at how translucent they are.
Yeah, beautiful.
Okay. Oh, sorry. There you go. You can see they're all. It's like an X-ray of the body. You can just. That's good size too. You know, 15, maybe 20, nearly 20 centimeters across. This one doesn't seem to be moving much, but you can just make out the trail behind it. You can see the lighter coloured sediment right there, right between the laser beams. Yeah. So it's moving upslope. Okay. All right, gents. Thank you very much. Yeah. Yep. We'll head on. Okay, we'll lift off. We're heading up our slope. This is the side wall of this very large U-shaped valley. The valley itself is around about 10 kilometres long. So the head of that is right up hard against the Ribbon Reefs. There's a shallow coral reefs, a barrier reef system on the edge of the Great Barrier Reef, and the head of the canyon lies just below that coral reef. So over the next 2 days, we're going to be sampling at different parts of this canyon. This is our deep canyon ROV site. Tomorrow we're going to work up at around 600 meters depth. Yeah. Okay. The middle part of the canyon. So we're at the lower part of the canyon now. We're going to work up on the middle part, probably around about 600 meters depth. We expect it'll still be muddy, but I mean, one of the things we're looking for is exposed rock that can tell us something about the origin of these canyons. So far, most of what we've seen has been mud, sandy mud. Oh, look at that. That's a great view. Cynobranchus again, perhaps? Yeah. And then, and what will be really interesting, come Monday, which is in 2 days' time, we will be right at the heads of these canyons. We're going to try two different canyon types. One of them is reef blocked. So in fact, the head of the canyon sits directly in front of a coral reef. So the sediment and any marine life that comes from there would rain down from a coral reef, a shallow coral reef that's directly adjacent to it. And another part of this canyon system that is completely connected to the shelf through an interreef channel. So lots of different kinds of sediments coming through there, quartz and carbonate material and all kinds of organics. So it's a different kind of canyon head. So in 2 days' time, we'll go and look at both of those canyon heads, and if we're really lucky, we may see like something coming up in the shadow. Oh yeah, look at that. Another big Benthodites. This is one of these big purple sea cucumbers we're just looking at. Just love that swimming action, eh?
It's the colour contrast with the ocean. It's just superb.
Yeah.
And we have seen these when they've been disturbed, where they've gone from the seafloor, just pushed directly up into the water column. But this one was just drifting along. look at that.
It's right on top of us.
Little white dots, probably part of the animal, not necessarily, you know, just stuck on pieces of material. So, yeah, but beautiful. Yeah, I wonder, Dougal, you know, we're talking about convergent evolution here, and this is a sea cucumber, a holothurian, that is, you know, swimming like a medusa, a anthozoan. So I'm sure you've probably contemplated this, how you get two different animals using swimming motions that are similar.
No. Oh, you don't have the program.
What's that? Huh? Hey, we're just going over one of those quite different sea cucumbers, pink, possibly Benthodites again. We did reach out to holothurian expert from the Museum of Victoria, and she identified 3, possibly 4 different species of deep sea cucumber from this part of the GBR. And this is from a vision that we collected from the ROV Zona in 2017, much more shallow than here, but similar kind of environment, fairly muddy. We only had the camera down for about 20 minutes. It wasn't great vision, but it was enough for us to say that, yeah, okay, this is an environment where you're going to find lots of sea cucumbers. So quite curious as to what different species are down there, because it's, you know, that's the name of this game here. We're trying to do everything. We're trying to both describe the geology, the marine biology, and of course we need samples as well. And Jeremy's going to take over the view here. We'll just see what's coming out of the seafloor here.
Gorgonian? Sea pen, maybe?
Or is it one of these mystery sponge spicule stalks with some kind of anemones growing up the side? What do you think?
Let's see. Oh, yeah.
No, there's definitely polyps growing.
These look like Gorgonian wrappers. So again, these
look at the white. See the white stalk down the bottom?
Yeah.
That would be like a hexactinellid sponge spicule.
Can we get manual focus?
Yeah,
let's see.
Auto right now.
I just took it off.
I know, but like if you get out of sync over here, then it goes all crazy. So
how about now?
It says it's on right now. Are you on or off?
Off.
All right. Now try.
So we're just trying to manually zoom in here, get a closer look. Now they can be a bit ambiguous here. So first time we saw this earlier in the dive,
Are you sure you're not backwards over there? So try your elevation.
It's the other way.
One more time. Okay. Now, are you clicking the button or pushing down on it?
Clicking.
Try turning it down with
it says it's on auto.
We're just playing with the manual focus here just to try and...
Oh, so it's just backwards.
Yeah, you're backward.
But that's fine. As long as we can.
There we go. Coming into view now. Yeah, I was just going to say they're a bit ambiguous.
Oh, beautiful.
So what do you think, Jeremy? What's going on here?
I mean, are these essentially parasitic
anemones?
Anemones that are colonial, growing on a dead sea pan or something?
But it's like a skin that's grown around the outside. You know, could they then pod as they go higher up the stalk?
Let's try to find a place where they're not. So they're the white. You can see.
Yeah. Okay. So in this part of the world we have these stalked sponges. They're siliceous sponges, and they have these quite distinct white stalks. What we think is this is the remains of a stalk with some kind of colonial anemones that are growing as like a skin around the outside with their own polyps, and of course they then pod off. It's like they're growing around the shaft of an old, a dead siliceous sponge. Would you go with that, Jeremy?
You often see on dead gorgonians, they call them gorgonian wrappers, where colonial anemones would just take over something that's coming out of the ground, and so dead gorgonians would be taken up by these things. And you can see how unorganized the animals are, so they're probably not coming from the colony. You'd imagine it would come in rows of 2 or 3 or 4.
James is saying probably anemones, maybe.
So they look like colonial anemones.
Can you get closer in?
No. Well, we can get closer.
I mean, no. I mean, just we were at max zoom, are we?
We're at max zoom.
Yeah. Okay.
We just check the top out.
We have collected something like this earlier in this dive, so I think that other one we got for James. So that might be. What do you think? Did you want to click a piece?
I don't think so. I think it's okay.
All right.
Yeah, we can get going.
Cool. Thank you very much.
Cool.
Leave it alone.
Yep. So still lots of surprises in this environment, even though, to, I guess, the untrained eye, it might just be a moonscape. But, if you look closer, there's all kinds of things. So we have these, just off in the distance, there's a big pencil urchin. They're pretty interesting. They're just going into the middle of the screen now. There we go. It's mobile, can move around. What's that? Is it a fish just off to the left here?
Goosefish.
Goosefish.
That's the first one we've seen on this dive, I think. Now, nice. Okay, Fish Army, get to work. What do we got?
Goosefish.
Goosefish. It's from J-Rod. He's seen more underwater ROV vision than anyone, so I won't take you.
Got a smiley face.
Yeah, no, grumpy face.
Grumpy face.
Yeah, hasn't had his coffee. There we go. We're just playing with the manual zoom. See if we can get a closer look. That's nice. Wow, look how.
Not a goosefish.
No. Coffin fish. Is that anything? Here we go. We got Diabranchus in the Atlantic. Wombat, yeah. Thanks, mate. We've already done that one. It is little anglerfish. Very possible. This is it's the first one we've seen on this dive. God, it's even got really distinctive nostrils, almost really comical face.
Looking like a dog,
Doesn't look impressed though, does it?
No.
If we could zoom in more, we can't.
We're at max zoom, are we?
Yeah.
Yeah, I have to agree. What a face. He's got that sort of intrigued look, would you say? Like
inquisitive, yeah.
Like, what the heck? Yeah. But what's strange is the skin. Look at the, not scaly like your classic fish, but, with all kinds of protuberances. Because there's plenty of fish that do this. Think of, stonefish as a classic, almost got a slimy outer body and have all kinds of things hanging off them. Oh, yeah, you're right. That's great. I'm not amused.
All right.
Thank you. That's great.
Yeah, really nice. And James was saying that the anemones might be something else. We'll have to look. We'll have to do some research on what we're seeing wrapped around those dead corals.
So just talking about data and the like, the Schmidt Ocean Institute is all about open data, and so all of the data that's been collected, both the multi-beam and the underwater vision data, the screen captures that have been taken, all of the oceanographic data, the sensor data, is open to the public. And we very much welcome people from ashore, from different research institutes, museums, to reach out to. Stalked crinoid.
There we go.
All right, now we need to look to see if there's other anthozoans on it.
Or could it be just something attached to the stalk of a coal sponge. What are we looking at
No,
here?
it looks like it's one piece. See how it gets larger.
All right, now, okay, I don't think this is a stalk crinoid. No, there's something different here. But boy, it's interesting. Okay, Jeremy, would you like to take over? This is your part of ship.
Yeah. Looks like a octocoral. Let's try to see the texture of this thing.
Wow, okay. So we'll have some kind of body.
I think this is where...
that's anchored into the seafloor.
Yeah. All right. Oh, we got a dust storm.
Umbrella.
Oh yeah. Okay.
Super rare.
So, yeah, we're.
Umbrella.
So this seems to be an octocoral. Let's look at the.
Long. What?
It's very long.
Half a meter. Weird.
Beautiful.
Weird sea pen group. Yeah. Let's have a look at the tentacles. Yeah, it looks very octocoral-like, doesn't it? Yeah. Not a feather star at all.
No. Yeah. Look at that. So you can see 8 tentacles.
Yeah.
I'll try to focus. Yeah. Oh, look at that.
That is really interesting.
So what we're gonna try to find a good view to just let it sit for about 30 seconds.
Can we try to get the light a little bit better on?
God, that is.
Yeah, if you think so.
Yeah.
Yeah, it's a beautiful octocoral. So our octocoral collaborators, Kathy McFadden, will help us with this, and also Andrea Quattrini, curator at the Smithsonian Institute.
What's going on there, Jeremy? There's something in the middle.
Here, let's take a look. I'll try to zoom in on. So behind one of those polyps in the back, facing away. We're just trying to get the light right. All right, I think. Oh, it looks maybe like a little, some sort of, maybe a shrimp or
an isopod, perhaps some kind of crustacean, maybe just attached.
Let's try to.
Like a little parasitic crustacean of some kind?
Yeah, I think I see legs on the side. It's going to be hard to see.
It's flat, so isopod-like.
That's the mouth. So you can see the mouth of the polyp, and then the 8 tentacles around it.
Does it have a polychaete in its mouth? Yeah.
Yeah. So there's a little crustacean. Yeah, of some kind. Parasitic crustacean, perhaps. Just using it as another home, another ecosystem.
Beautiful. All right, I'm going to zoom out so we could take a nice image with the scale, so we could kind of see how long it is. I'll put the.
That's 10 centimeters. You're looking over half a meter.
Let's see the 10. It's big. So I did it very long.
Two saying Anamura.
You need to look up more if you want to get it on.
So
look how long that is.
Yeah, to get a sense of the length, we're gonna have to put the lasers down at the level of the base of the. Okay, so that. So 10, 20, 30, 40, 50.
Yeah.
It's a half a meter in length. All right. Let's grab it. Whole thing. And what is it attached to is a good question.
It'll be another one of those nautilus shells.
Yeah, nautilus. Yeah.
We hope. See,
come back on with all the lights.
Yeah.
It'd be hard for you to beat that.
Absolutely. Yeah, so it has a peduncle, right? So
so it's anchored into the seafloor in some way. They have to. It's all over otherwise.
So, yeah, it's big. It's about a half meter in length. Muscular peduncle buried in sediment. So it's not attached to a rock. we're about to see. Sometimes, yeah. Interesting. Yep, so earlier on in leg 1, we collected a black coral attached to a nautilus shell in an area otherwise impossible for a hard coral to, or a black coral to find substrate to grow. So this... Oh, right. Yeah, so we'll have kind of roots. Yeah, okay. Yeah, right.
That's what you wanted, right?
That's exactly what I wanted. It'll go in box 3-4, of course. Beautiful. Now the challenge here is, what do we do with it?
You can see the base right there.
Oh yeah, look at that. See how wide it is?
I'll just zoom in.
It's got a root, track root carving.
Yeah, we'll cut a little bit.
Yep. So, well, it just looks like
Like a big stalk, like a big arrow in the circle.
Yeah, it doesn't seem to have. I thought maybe it would have like kind of roots that would kind of dig into the sediment, but it seems pretty.
But it's a good distance in.
And I mean, it's a very hard substrate. Maybe it doesn't need much to. You can take over.
So talking about octocorals here, they don't have this full hard skeleton like you find with other scleractinian hexacorals, but they've got spicules embedded within flesh, so it gives them some rigidity, doesn't it?
All right, can you catch it before it hits the ground?
The current is pretty strong here.
Wait, are you rewinding? It looked like it was amazing.
How's that?
Oh,
well done.
Unbelievable.
Sign him up.
It should have a hard axis. You okay? Yeah, James. So when we get into the lab, we'll take a look at it.
Well, I didn't have the phone. I just grabbed the button.
So, James, if you want to be part of this, you want to actually have a look at some of these samples, you're welcome to do that. You just need to reach out to Chief Scientist Brendan Brook, and you'll be able to coordinate who you actually need to speak with. So that's a big part of this expedition is to be able to bring in all the world's experts who can help us identify animals. So you're very welcome to get in touch with us.
Oh yeah, if you talk to Kathy, James, she's who I would ask. So, yeah, absolutely. Yep, sure. Yep, so all of the corals that we collect are going to go to either the Queensland Museum or the Museum of Tropical Queensland, so either in Brisbane or in Townsville. And we're going to take the tissue sample of all the specimens that we collect. We take tissue samples and we preserve the rest of it in 75% ethanol. So we'll be able to sequence, and if Kathy's involved, which I'm assuming she is, and she's going to be interested in the specimen, she'll be using conserved elements to add this species, if it's not already added, to her phylogenetic tree to understand how this specimen evolved, how this species has evolved, and its place in the order.
So the question is, do you have to be an expert to get info on what we find? The short answer is no. The longer answer is that it's going to take time, of course, to identify lots of these animals. Hence, we do actually reach out to a lot of the world's experts to do this, because it does take a bit of a specialist knowledge to kind of build up that knowledge. But, beyond just taxonomic identification, even at the very early stages, we can start to explain the habitat. So we're talking about this classic muddy environment. We understand quite well the water column and the properties of it. And so over time, we'll probably do scientific publications. In fact, we've got all kinds of other things happening. There's a lot of outreach going on, both on the internet and also through vision. There's actually a documentary being made with this expedition right now through National Geographic. So, there's lots of ways that people can can be part of this, both during the voyage now, but also afterwards as we really start to drill down and understand better all the different parts that we're looking at.
Yeah, there are very few specimens that we actually identify to the species level ourselves because everyone really focuses on one group. So, besides the black corals, all the other specimens, I'm going to be sending images and subsamples to others who are working on these various groups. So, we welcome, and collaboration is the key. It's the only way to really understand how these different anthozoans have evolved.
Yeah, exactly. Science is all about collaboration. No one person can do it all. And so, my time in academia, it's quite clear that the best scientists are the ones who can reach out and build the biggest teams. And, in this voyage in particular, we have all kinds of people. We have engineers, robotics engineers. We have the coral experts. Jeremy is focused on black corals. We have quite a few marine geologists here with lots of experience. We have oceanographers, we have Dougal with his plankton. And that's just the start. And so, if I, quote the founder of, the Schmidt, Wendy Schmidt from the Schmidt Ocean Institute, she talks about these voyages as being the gateway to knowledge. And really, we're just at the start of this. And so, that knowledge will just build through time. And, it may take years to find out names of animals and the like. But straight away, we're getting an insight into the deep Great Barrier Reef, which has always been a real goal of mine. And it's just thrilling to be able to get down here with the SuBastian and the ROV Falkor. Because this is quite tricky. You see, you got this
current
long animal. And
it's got a lot of drag, and the current is very strong.
Yeah, that's right. Current's been a bit of a battle all the way all today.
Luckily, we have J-Rod here, who not only caught it floating in midair, but is now fighting the currents to get it in the box. What a great find. It's amazing.
Yeah. Yeah, it's beautiful.
I actually have
but no shortage of food. You look at how much we're just sitting here, and you can see this marine snow drifting over the landscape. There's a lot of food here, don't you think, Jeremy?
Well, yeah, and if this can attach to this type of substrate without any sort of without a rock or anything to attach to any hard benthos, why aren't there more of them in the area? Why isn't it a field?
Yeah, indeed.
Yeah, good question.
It's a case of recruitment, isn't it?
Okay, close it a little bit.
Well, I guess we'll see now. This might be, as we get a little shallower, maybe we'll see many of them.
Yeah, and as we see the rest of the canyon will build up as well.
Checklist.
Local variation, different aspects, maybe.
Right.
Yeah, so this is a systematic survey of this canyon. We're at the lower end of it, the deeper part. Clearly it's very, very muddy. But over the next couple of days, we'll go into the middle canyon section, up to about 600 metres, so could be very different marine life there. And ultimately, we'll be at the head of the canyon, probably around maybe 300 metres of water, so relatively shallow, mesophotic zone. It's just still very tricky. You almost wish you had a second arm that you could grab that bit to hold it down so you could push that bit down.
Well, they do have two arms. Can you...
Yeah, I'm going to try that.
All right.
Turn around.
This is a first for me on the Falkor. Both arms are being utilized to grab this coral.
It's like the moon in the background.
Yeah, grab it by the floaty top. That's the scientific term, by the way. The floaty top. This is going to be fun to photograph.
So one of the questions on Facebook, what's the temperature? So the
3 degrees.
3.1 degrees C, so just a little bit above freezing temperature, very cold. If you picked up this mud in your hand right now, it's not quite ice cream cold, but it's getting close, and it would definitely feel cold in your hand.
Seems like it should just fall right in, right? They just need to get it from the left because the current's coming across the right.
Yeah, so the way we're going up the south side wall of this big valley, the canyon, and we've got the currents flowing into us. So that's why it's all a bit awkward.
Yeah, we'll have to call up. 5:30. Well, they've been called.
Yeah, as late as possible.
Yeah.
So this is definitely the hardest specimen. It's just so positively, it's not heavy, of course, at all. So the current is just picking it up like a parachute.
You can't close the jaws. You can't close the boxes that are try.
All right, they're attempting the two arm.
Alright, Adam, so what I'm going to do is I'm going to push this down.
Yeah.
Out of the way of our floor.
Yeah, we don't want to grab the.
I'm not grabbing.
We don't want to destroy the tentacles and the polyps, which makes it very difficult because that's the part that's floating. Oh boy.
That's that like crustacean we saw still hasn't let go.
It's a butter or whatever it is. Yeah. Oh, we'll definitely preserve it.
God, it's really not playing the game, is it?
Maybe just hold it in your arm the rest of the dive. Like a balloon.
Tricky.
Could you put it in the coral tube? Is that a tube that you have now?
Yeah, it's going to be just as tricky.
It'll be hard.
Especially with it blowing. It's just the current's coming right across, so it's just blowing it.
Right on.
Basically the exact wrong area.
Just a touch.
Someone equated this to getting a cat in the carrier to go to the vet.
Mate, I know all about that. I just took our cat to the vet yesterday, or the day before last. And, it was like that. I am not going to get that needle.
I'll just leave it like that, actually.
If you can,
Leave it close.
if you can just hold it in place, it'll be all right.
Yeah.
Hold on, let me try something else on.
As long as you get the head in, I guess that would be obviously the. Oh yeah.
Alright, I'm going to coast it into this area.
Oh, I see what you're doing. Yeah. Oh, very smart.
Clever. There you go. Right.
Jam that bit.
It's like playing pool.
Yeah. What a difficult one.
Yeah, the big ones. The big floaty one.
Yeah.
Stay in there.
See the head, we'll call it the head, is still coming out the side.
Damn.
Octocoral Tetris, that's good.
Take it under closed lid. Nice.
Well, this is definitely problematic coral of the day.
Worth the effort, though.
Boy. I imagine the stalk is pretty resilient. If we close it on the stalk, I think it'd be fine. And at this stage, I mean, as long as we get the tentacles and the polyps in, we're good. All right, now we're going to attempt to close BioBox lid 3. Nice. Yeah. Yeah. Oh, yeah. Good. All right, we got it.
Yay!
Thank you, J-Rod.
The turtle's happy.
J-Rod's saying this one better be worth it. All right, so with the time we have left, we're going to continue up the side of this canyon. We spent a lot of time collecting lots of really interesting specimens and observing the what's living on these seamounts from the base, and then just the base of this canyon, and just a little bit up, less than 300 meters. It just shows how much there is to see, and in a habitat that you would assume, not much lives there. And it's true, there are large areas where there's not much occurring, but you look close, you'll find stuff, and then of course you see black corals and these octocorals.
Or that fish we just saw. It's strange.
Yeah.
Lots of little surprises.
Yeah, real spectacular.
There's a chance we see something really cool, so we'll let you know.
Yeah, of course. Of course. Oh, what's... just wait 1 second. I just want quick zoom in. At this white... Okay. What is that?
Sit down and look at it here.
ROV Bridge, do you have an update on the ETA to the surface or Leo?
That's so weird.
Are y'all gonna wanna look at Christmas tree?
Yeah.
Give me just a quick second.
Let's just...
We just don't need to finish the transact. So
Are y'all good for?
Want to be on deck at 5:30?
No. We're talking about leaving bottom at 5:30.
Okay. Yeah, that's fine.
So this is.
You're okay with that?
I need to see how many tentacles are on these polyps. It's a really odd. You can see it's got 4 rows of branches. It's small, and the bottom one's the longest, and they get consistently smaller. So let's see if this is a black coral. This is a super weird black coral. And wait for the dust to settle. No, it looks more like an octocoral. Wait, let's see.
Oh, with a couple of brittle stars at the base.
All right, let's focus.
We're ready just to feed it into the sections.
Almost with that rough.
I can't see.
Right on that film. I want to look at which icons that contrast this there.
Can I get manual focus? Is it on manual?
Oh yeah, here I got it.
So
It's on manual.
Okay. I think. Yeah, could this be a sponge? You think?
Doesn't look like a sponge to me. Looks like some kind of octocoral.
It's really hard to tell. I don't see a polyp and tentacles. I just see protrusions. And look at the base. I can't zoom in anymore, and I hesitate to go closer.
I can bump up a little bit.
Way to not stir up the sand.
The good thing is the current's kind of going across from us, so even if I'll just try to slide it.
All right, let's do it. This is something that we haven't seen. I know I want to get up as well, but it's a really interesting. Yeah, it doesn't look like an octocoral. I can't see polyps and then tentacles coming off of polyps. All I see are these small protrusions which look like tentacles, but without a polyp, they're something else. There's a
brittle star swimming away from it. So it's using its forearms to row.
yeah. Let's take a close look. If it is a black coral, I couldn't tell you what kind of black coral it is. I mean, it has 4 rows of branches that decrease from the base to the top.
Sorry about the viz, but it should clear right away.
We're closer?
Yeah.
Okay. Yes. It's small. It's very small. The whole thing is just 10 centimeters across. Really interesting. All right, we're a bit closer now. Let's try to zoom in.
So no, it's manual.
Are you able to focus it?
No.
Try now. There you go. It's over.
Yep. All right. So what's interesting is the red seemingly underneath the tissue is this red color. So this wouldn't be a black coral if yeah, they look like spicules. I'm gonna go with the sponge as well. And this is really interesting. Let's grab it.
Want to sample it?
Yeah, absolutely.
Sample.
Yeah. Yeah, if this is a carnivorous sponge, we're making a certain curator at the Queensland Museum very happy. We actually described the new species of carnivorous sponge in the Coral Sea last year. Those came to a maximum height of a few millimeters, 5 millimeters. So this one's much bigger, and it has a much different morphology, but a lot of the carnivorous sponges are undescribed.
Where are you going to want this to go?
1B is empty.
1B. Yeah, it should fit there, yeah.
Put this on to set.
Then you're transcribing these into a
Have no.
Yeah, thank you all for your comments on helping identify it. Yeah, please. Yeah, we'll see. We'll see how brittle it is when we pick it up. You can see how small the colony is. It's very intricate for being so small. Well, I guess size doesn't really relate to how intricate it is, but it's always amazing to see how much detail is in something so small. Yeah, feel free to dump the sediment there with it.
You want a closer look at this one?
No, it's all right. Oh, you could see. Yeah, it's okay. This one should be a bit easier than the last one to get in the box, I'd say. Yeah, it's beautiful. That's a good one. That's a good one.
Full evening, Hills.
All right, thanks, Sherrod.
Yeah.
Is Merrick not around? Virtually, of course.
Yeah,
That's a cool one. All right, this area is so interesting. You know, we thought we would find weird life, and that's exactly what we found. You know, various weird from different groups, lots of different critters. It's spectacular. Thank you, Narita. Nice to see you on here. Welcome aboard virtually.
One right here, one up there, one right there.
The same thing?
Well, this one is
Oh, another one. Another one of these.
Yeah.
Another one is
Top right,
Gorgonians.
or top and center of your screen.
Yeah, we'll keep cruising up to see what else, but it is amazing. So we asked the question, why aren't there more of them in this area? And the answer is there are. You know, we just came across the first one. So the question is, will the live stream continue during the ascent? We tend to turn it off when we're bringing it up, but we'll keep it on as long as we can for you. Yeah, amazing.
5:30.
Just a big holes to it.
Yeah. Seems to be a sort of depression ahead. Oh, that's a pencil urchin.
Yeah. Seen quite a few of them on this trip. Not so many trails coming in front of them. Sitting there quietly.
Lazy.
Because they're completely mobile. They've got little tube feet underneath. They can push out, like the hydraulics can just push them out and move across the landscape.
Yeah, so you see lots of little holes, little hidey holes.
And these strange clusters of burrows or holes in the seafloor. Very curious what might be making them.
I wonder if we could... something. That's okay. Yeah, we'll continue up. So,
we got Chris Jones joining us. G'day, Chris. Chris works for the Marine Park Authority here at Great Barrier Reef, and you know Chris? Yeah. So he runs the Eye on the Reef app that you can download. Now, these big burrows, my guess is these belong to giant isopods, so that's what I'm putting my money on. But we haven't seen any giant isopods on this ROV dive, but we have on previous ones. They have that distinct flat-shaped floor with a sort of a domed roof. Yeah, we've seen these before.
Yep. Yep. So these must be sea pens, or I guess glass sponges.
Orange tubes that are laying on the ground next to them. I've seen them a couple of times now.
Orange tubes.
Down to the left of it, at the base.
Is that a tusk? Is that a tusk shell? No.
Yeah, Dougal's saying colonial anemones again. Yeah, that's what it looks like. Growing up, the stalk of possibly a sponge, the stalk of a stalk sponge. The head of it's long is dead, and it's just left this long siliceous sponge spicule behind, and some kind of colonial anthozoan is growing up around the outside.
You want manual or auto right now?
Well, I'm waiting for the dust to settle, and then I guess manual because it's very thin, small. It's on manual now. Oh, there it is.
Switch your hand or switch it right back down.
Okay. One focus, and we'll know. Yeah.
It should be a manual right now.
All right.
Just good starting point for it.
Yeah. Well, it looks pretty good. So for me, the telltale sign is that you could see the kind of the tissue beneath these, what I'm going to call colonial anemones, and where there aren't, you could see what's beneath it, which seems to be that white stalk, which could be whatever it is, whether it was a soft coral or a glass sponge. And then as you go up higher, you see more of these, without much order, these anemones protruding. If it were a part of the colony, they would be more consistent. There would be rows of them, not colonial anemones. You think it's a sea pen? I'm not sure.
How long is this one?
Because we're running out of time.
Yeah.
We've only got about 20 minutes left.
Yeah.
So let's see what we can cover in 20 minutes.
Yeah. Sorry, we have to continue moving on. We have under a half hour to go before we need to reach the surface. Yeah, I'm not sure, but we did collect one earlier on that I think was similar to this. I might be wrong, but I'll take a look at it, and it'll be pretty obvious when we put ethanol on it, whether the animals leave the, see how they react.
Sea cucumber. Yeah. Bent over. And you can see its internal, its intestines there.
Another sea cucumber, yeah. All right. Yeah, we'll keep moving on.
Strange clusters of burrows, holes. Yeah. They're clearly clustered together. Very obvious here, aren't they? I'd really like to know what's making those.
One organism doing the same thing, or
is it? It's just multiple feeding holes, breathing holes. Is
when you look at the trace fossils, you get branching structures.
How would you answer that question? Would you put a camera down and just record for days?
You could, but I saw Rachel Brzezowski before I came on here, and she was saying she was on the Heritage trip, and they actually put push cores in it. They were trying to try and capture the animal, and it came up nothing. So it's because some of these trails are really obvious what they are, but others,
what some geologists have done, but it only works intertidally, is you pour a resin mix into the burrow structure. It solidifies.
Sitting up on his legs.
Oh yeah, beautiful shrimp. Good big shrimp down in the deep. Yep, anemones. It's a good question about the pressure and how it affects the specimens that we bring up from the deep. For fish that have
burrowing anemones.
Oh yeah,
like a tube anemone.
Yeah.
Like if we touch that, it would probably zip straight back into its hole.
That's beautiful.
Yeah.
Just gonna come right up to it there, James.
That's okay.
The picture you can.
That's okay.
Yeah.
The question about specimens, bringing them up. So most of the things that we collected are corals, and so the black corals are made of a protein called chitin, and there's no pockets inside. It's very dense. So bringing it up, it won't affect the volume. So things like fish, if you bring a fish quickly from the deep to the shallow, it'll kind of implode, and that's why you get things that look like blobfish that look really ugly, but actually they're not ugly shaped fish. It's just the pressure changed so drastically that it made them look different than they normally do. So no, none of the things that we're going to bring up are going to change because of the change in pressure.
Poor blobfish, it's such a bad rap.
Yeah, poor blobfish.
Must be truthful.
Well, we collected an amazing number of specimens today. All the groups that we were targeting, except for Sclerotinia, but that's okay.
They'll get their chance.
Yeah.
In the next few days, we're going shallower. Right, each day we're going a little bit shallower until on the third day, probably Monday, we'll be at the heads of the canyons, sort of more mesophotic zones. So there's very, very likely we'll have opportunities to get some corals for Tom Bridge and people from the Museum of Tropical Queensland.
Yeah. It was a question about the temperature. It's 3 degrees. So it's actually interesting. When we first got down to 1800, it was 2 degrees, I think. So it really only increased by 1 degree. But of course, as you get closer to the surface, that rate of change of temperature increase will go up.
Okay, one of our eels. Just quietly sitting there.
Yeah, they don't seem too bothered by this monstrous.
No.
Anemone?
Sea cucumber. Perhaps. No, don't know.
So that fish we saw earlier looks like a halosaur.
Yep.
Thanks, Dougal.
Eel-shaped fish found in great ocean depths. Yep. So we appreciate people from the shore helping us ID these animals.
What's this?
Previous
Go look at that for a second.
expedition we had Fish Army joining in.
Just quickly to see.
Great help identifying all the different, mainly the mesophotic fish we were finding.
Yeah, we'll just look at this and then we'll. Yeah, it's okay. It's another small colony that is really hard to, catch in your eye. It doesn't really stand out. It looks like it has a crustacean hanging on it. Maybe it's a soft coral. Let's see. Interesting.
You want manual?
Oh yeah, please.
There you go, you got it.
Look at that. Oh. Yeah, look at that. So, it looks like a soft coral with a little crustacean hanging onto it. It's really hard to tell. It's pretty complexly branched. Doesn't seem to be much order to it. I don't know if you'd call that a bramble colony. There we go. It's weird. We haven't collected anything like this yet, so maybe we add this one to our collection.
See the shrimp? See these arms?
Yeah.
The squat lobster.
Yeah. Let's take a nice picture of it.
And it's coloured exactly the same.
Yeah.
As the.
There we go. So yeah, it's definitely a soft coral. This will be another one for Kathy to help us identify. It looks like it has like nodes almost, but not everywhere. Yeah. Could it be a bamboo?
Bamboo coral. It might be. Yeah, you're right. It might be like little nodes. Yeah.
All right.
It's a bamboo coral.
So Andrea Quattrini has been dying to get something like this. So we are certainly going to put this in, and then we'll call it a day.
Yeah, thank you.
Thank you. Yeah, this is a good one.
Yeah, cool.
Yeah, bamboo. You tend to think of bamboos as the the stem, the unbranched kind of bamboo-like, but
That's right.
this looks like it has those interchanging nodes of gargoynin and calcium carbonate.
Just a lot more delicate looking than what we've seen.
Yeah.
You know, you think of the bamboo corals of certain coral sea, they were, you know, long feather, really robust stalks, you know, like internode pieces like thickness of your finger.
Right. Oh, this is very thin,
delicate.
There's a small colony. And you could see that the branches are all coming from the same point on the stem.
Okay,
I don't know if that's consistent, but if it is, that would be a characteristic of the species. I'm no bamboo expert, you know, but corals tend to work that way.
Put it in C.
C, please. Yeah. Oh, we did good today.
Yeah.
I mean, this is a diverse area. We you know, we collected a lot of different things. This wasn't.
In faunal diversity as well. I'm sure Rachel will be impressed.
Yeah, lots of traces down there.
Yeah.
You know that rock?
Yeah.
That change of that's interesting. We have seen, we know it.
That was probably in place.
Oh wow, amazing!
I'm really pleased we saw that rather than just simply.
Indeed, yeah. It's hopeful. There's no sense of it. It was vaguely better, but not strong.
You can see it has like a root structure at the end.
Is it too big?
Ooh. What do you guys think? Too large?
Is it too big?
No, it's just hung up there.
Okay.
Keep pulling it in the boat.
Yeah.
If not, I'm sure it's no match for our hydrauligram of the wild buckskin.
That's a good question. What do we do with the associated species that come up on the corals that we collect? So what lives on the corals is a really important question to answer. Sometimes you get undescribed species living on undescribed corals, and you know they associate with that specific coral. So yeah, we have collaborators that are going to work specifically on the associated species. And in this case, crustaceans, we have crustacean people that are going to help us identify the specimen. All right, it's in. So you can see it's got some sort of a root structure that allows it to, attach to this muddy, dense sediment. And I guess, you ask what type of corals are going to live in these habitats. These are the types of corals that are going to live here, ones that can survive without a hard substrate. J-Rod is interested in what this is.
Probably just a stick. Is that everywhere we've sampled? Are tube-worn?
It's not bamboo coral like.
No, it's a tube, so it's hollow on the inside. So that's weird. I don't know. Do you want to throw it in the bio box and look at it?
No, I mean,
Okay.
yeah, I just was curious. They've been around every coral I've sampled.
All right, so I think we've exhausted. I think we're run out of time. I want to thank everyone for tuning in. It's been a really interesting dive. We collected great biodiversity that categorizes the things that live in these habitats. And like Rob said, this is the first time we're really getting a camera down to see what's living here. We have the area mapped really well, but what lives in this habitat? There's a connection between, the substrate, the habitat, and then the biodiversity, and making that connection and understanding what allows these species to live in this habitat is a really important question to answer. And it really comes back to how do we protect biodiversity using surrogates like different substrates and different habitats.
Tune in tomorrow morning for more.
Same throw time, same throw channel.
As off it goes. Right.